Innovation has many faces, and ideas can come from any direction. For example, Newton, he found himself below the tree and got an idea. Archimedes found that in his bathtub. Sir Gregor Mendel in his monastery. While many of us might get it today, right here in this poster presentation session. Welcome to Biotech poster presentation session. Today, as we initiate one of the grandest events in the history of Biotechnica, I welcome all of you to this virtual poster presentation session, where 30 contestants will compete to win the top 3 prizes. Excited? I am. Now, exactly 50 days ago, on 5th of August 2020, we had announced the Biotech poster presentation on the top 10 emerging areas in the BT sector as identified by the Government of Karnataka. Now, this is being done in collaboration with KTech, of course, Government of Karnataka, and ABLE, Association of Biotech-led Enterprises, and 5,500 students, more than that, registered for this event. 1,000 plus posters were submitted, and they were so wonderful. They were so creative. Trust me. They were, at one point in time, so much creative that our jury members had a tough time selecting these posters. But as you all know, it's a competition. So they had to, select someone, and someone has to win, someone has to lose. So we selected 133 posters that were kept for display. Right now also it is on display. And today, 30 top posters will be getting a chance to present it live. Also, we have opened the voting lines for the most popular presentation or the posters category, and it will close at around 5 o'clock today. So you still have time. Tell your friends and peers and family members to vote. And today, as I come to you live, I am joined by seven eminent personalities from the BT industry who will be presiding over the event and judging this event. So today I would like to invite Dr. Taslim Arif Syed. He is the CEO and Director of Center for Cellular and Molecular Platforms, as popularly known as CCAMP, Bengaluru. I would also like to invite Dr. Satya Dash, and he is the board member, NCL Venture Center at Pune. It's a Pune-based technology business incubator that supports early stage startups in R&D and science-based startups. And he's also a founding head strategy of BIRAC. I would also like to invite Dr. H.S. Subramanya, Director of Institute of Bioinformatics and Applied Biotechnology, IBAB, Bengaluru. And I would also like to invite Dr. Raja Mugasimilingam, and he is the CEO and founder of Genotypic Technologies, Bengaluru. So these people are going to be our judges. Followed by that, I would also like to invite Dr. H.E. Shashidhar, CEO of Cultiva Agritech, Bengaluru. Welcome, sir. And I would also like to invite Dr. Ramesh Bhatt, who is professor and head of the department, head of Institute of Agri-Biotechnology, University of Agriculture, Agricultural Sciences, Dharwad. And followed by that, I would also like to invite Dr. Kshitij Acharya, one of our eminent faculty members and scientists, transcriptomics and reproductive biology at Institute of Biotechnology and Bioinformatics, IBAB, Bengaluru. And now that you can see all our judges on the screen, thank you, sir. Thank you for taking out your time from your busy schedule and joining us today for this mega event. All right. So now that we have all our judges on the screen, without taking much of your time, let us begin the presentation. Shall we? Yes? All right. All right. So welcome, everybody. Welcome to the poster presentation session. I'm your host, Shekhar, and now I'll be bringing up all the judges on screen. And our first poster presenter, Mr. Karthik Gehlot, will be joining us shortly. Now, in the meanwhile, I wanted to urge you all to share this live poster presentation as much as you can with all your friends and family members. Bring in everybody because you're going to witness some of the most intellectual talks in the industry right here live. Okay, so let us bring in all our judges. All right. So there we go. Welcome, judges, to the session. I hope everybody is audible. Yeah. All right. So we will start our presentation session. Before that, I would like to urge our judges to speak 2 lines each and share their thoughts before we begin our session, starting with Dr. Satya Dash.
Well, thank you for having me here. And I think it's great to see so many students really interested in biotechnology and in new areas of biotechnology. On behalf of the judges, as we were talking, my personal interest is to see, how you are planning to take the idea that you are presenting in the poster into the future. In whatever way you want to work with that poster, whether you want to do a PhD, a postdoc, build an enterprise, Dr. Raja is here. But I need to understand what you are thinking about the idea that you are presenting and how we are going to use that idea to take it into your future. Thank you for having me here.
All right, there you have it, Dr. Satya's viewpoints. Now bringing up Dr. Raja, if you could share your viewpoints before we get started quickly.
Yeah, thank you. This is a great event. First time I'm actually attending an event, a live event where posters are being presented instead of taking a notebook and going and seeing the people. The pandemic has made this. See, I'm going to only look for any of the posters which has a good business potential. Need not be even your idea. It can be someone's idea. Can we sell it? This is the only thing I'm going to look for in your posters. Thank you and good luck, all the presenters.
Okay, there you have it, Dr. Raja. Proceeding ahead, Dr. Shashidhar, if you would like to come in and share 2 words with us.
Yeah, it's a pleasure. First time for me also, like Raja. We are used to moving around posters and asking questions and looking up and down, and so it's a new experience. It's nice. I want to see how the future of India stands with these bright young minds. From
Right.
5,500 participants, 1,000 posters, amazingly come down to 30. It must have been a great job, difficult job to
Yeah.
bring down to
Of course.
30.
Right.
So congratulations, and let's see what we have on the table. Thank you.
Right. All right. So there you have it. So we have Dr. Kshitij Acharya also from IBAB. Dr. Kshitij, if you would like to come in and share your viewpoints quickly. Your mic is muted, sir. You need to unmute your mic.
I'm very excited to hear most of the students who are presenting. I don't want to say much, but I would like to congratulate the organizers, particularly the Karnataka government, KBITS department, and Shekhar for arranging this and giving us this opportunity. Really looking forward to hear you all students. Thank you.
Thank you, sir. We also have Dr. Ramesh Bhatt. If you could pitch in quickly, sir, if you can come in.
Yeah, thanks for the invitation, as well as I'm really happy to be here. I've been mainly looking for the originality of the idea and the way
Right.
how the idea is implemented in terms of carrying out the experiment and generating the data. Thank you.
Of course. Yeah. Thank you, sir. Also, we do have Dr. Subramania. Sir, if you can come in and share your 2 words before we get started.
Okay, thank you. First of all, thank you very much for inviting me
Also, we
to.
do have Dr. Subramania. Sir, if you can come in and share your 2 words before we get started. You're getting an echo. You need to.
Are you able to hear me?
Yes, we are able to hear you.
Okay. So first of all, thank you very much for inviting me to participate in this event. I think it is a humongous effort and very motivating for students to participate in this and present in this event. I think have really put in a hard to select the top posters for this presentation. I'm really looking forward to see some. But unfortunately, because of some deadlines with DBT, etc., I may not be able to attend to the fullest extent. But I will try to stay as long as I can. Okay.
No problem, sir. It is really a pleasure to have you here. Thank you so much. All right. So now I would like to quickly bring in the presentation part, starting with Mr. Karthik Gehlot will be right here, and we'll start with the presentations. All right. So Mr. Karthik Gehlot, there you have it. You can start with your poster presentation.
Okay. Good afternoon, everyone. Myself, Karthik Gehlot. I am from IIT Roorkee, and I am presenting my poster on bioethanol production from lignocellulosic biomass by co-fermentation using Zymonas mobilis and Piscia stiphilis.
You need to be louder.
As we all know that bioethanol
You need to be
production...
louder.
Hello.
Yeah.
As we all know that the
This is better.
bioethanol production from the lignocellulosic biomass is a very promising technology nowadays, in which we are using combination of 2 microorganisms to carry out the ethanol fermentation process in the bioreactor. So we are using Kahn's grass biomass as a lignocellulosic biomass, which can be hydrolyzed using acid hydrolysis technology into glucose and xylose monomers. So lignocellulosic biomass is basically a complex polymer of cellulose and hemicellulose, and cellulose is corresponding to the glucose monomer, and hemicellulose corresponds to the xylose monomers. So we carry out the combination of both these organisms in a bioreactor, in which we ferment simultaneously both the sugar in a sequential manner. So for the glucose fermentation, we have Zymonas mobilis, which is a anaerobic bacteria, and for the xylose fermentation, we have Piscia stiphilis, which is a aerobic organism for carrying out the xylose fermentation. So this is the overall picture of our bioethanol production process, in which we are taking the lignocellulosic biomass, then we are using dilute acid and steam to hydrolyze it into the glucose and xylose monomers. Then these monomers are taken into the liquid form, and they are very acidic in pH. So they are being neutralized to around 7 pH, and then they are taken into the bioreactor. Then we set all the fermentation condition around 30 degrees centigrade is the temperature, 5.5 is our pH, and then we carry out the bioethanol production process. And then after the completion of this process, we carry out the distillation process to separate our ethanol. So basically, there are so many lignocellulosic biomass available to carry out this process, but we have selected three mainly biomass in our lab: Kans grass biomass, wheat straw biomass, and bagasse biomass. So Kans grass have around 44% of cellulose content and 21% of hemicellulose content. So which is a very high content as compared to wheat straw and bagasse, which have around 39% and 37% of cellulose respectively, and 25%, 26% of hemicellulose respectively. So next slide, please. So in this process, we have first figure show the fractional hydrolysis process that we have column in our lab in which steam and combination of dilute acid to carry out this process. So the complex chain of the cellulose and amylose broken down into the glucose and sugar monomers, and this is taken into the liquid form in our vessel. The vessel can be anything. We have taken our hydrolyzed in the flask. So this first picture of flask show the pure hydrolyzed in which we have glucose and xylose, both the sugars. So when they are get fermented using both the organism, their color changes from dark brown to slightly yellowish and brownish in color. So this process can be done into the flask level also and bioreactor level also. So here we have optimized the process in first the flask level, then we have carry out the process in the bioreactor process, and this is our bioreactor setup that we are having in our lab to carry out this process. Next slide, please. So in the next slide, we can see this is the trend of our the fermentation process that goes around during the full process. So Zymonos mobilis ferment the glucose into ethanol, and it takes around 16 hours to ferment the glucose into ethanol. And we have two types of media here: synthetic and hydrolyzed. In synthetic, we have artificial glucose and xylose, and in hydrolyzed, we have natural and glucose and xylose. So in synthetic media, Zymonos mobilis is able to give around 97% ethanol yield efficiency, which is around 0.49 gram per gram, and this same can be made on the hydrolyzed also. Here also we get about 92% efficiency on hydrolyzed also. Similarly, in the case of Pisha stupidity, which ferments the xylose sugar in the synthetic media, it is able to give around 94% efficiency for the ethanol production, and the similar efficiency is produced on the hydrolyzed also, in which we are getting around 90% efficiency, which is comparable in both the synthetic and hydrolyzed. So this technology is very much promising for the bioethanol production on hydrolyzed. So we are now in the next process. We have combined both the in both the sugar in the single vessel. In a single vessel, we have taken glucose and xylose. So both the sugars are fermented in a sequential manner. First, the glucose is fermented using Zymonos mobilis. Then xylose is fermented using Pisha stupidis, and we are able to get around 0.468 gram per gram ethanol yield coefficient, which is around 91.5% of the maximum theoretical yield. And when we have compared this value with our literature, we are able to get that the ethanol productivity that we are getting is around 1.56 gram per liter per hour, which is very good in compared to the literature which is in the existing technology. Existing technology are using around 40 to 60 hours to complete the whole process, but our process is taking only 30 hours to complete the process. And in this technology, we are also using the distillation process, which is used to remove the ethanol from the vessel. This ethanol can be inhibitory effect on the yeast species, which is very helpful in removing the inhibitory effect of ethanol also. So distillation process is very much optimized process for our this condition. So this process can be taken out at the pilot scale also, and we are trying in our lab to get this technology at industrial scale so that the whole country get benefited by this process, because it is very low cost process, and our ethanol will be costing around 30 rupees per liter as compared to petrol, which is existing and which is very expensive. So I think this our technology is very promising, and it can be carried out on large scale also. We have carried out this process on 7-liter bioreactor, but it can be done on 50-liter, 100-liter, or even 1,000-liter bioreactor also. So this co-fermentation technique is very good in terms of high ethanol productivity, and it can be done on synthetic media as well as natural hydrolyzed, and we are able to replicate the results on both the types of media. So yeah, that's it.
All right. So there you have it. Karthik has finished his presentation. Now over to the judges to ask questions.
So I had a quick question, Karthik. What are the problems you see you can think of when you scale it up?
Okay, the major problem in the scaling up is the hydrolyzed concentration. In the at the lab level, we are getting the hydrolyzed in very low concentration. It means if it is having 44% of cellulose, then it should give 44 gram per liter around glucose, but that we did not get. We get very less sugar, around 8 gram per liter when we hydrolyze around 10 kg biomass. So this is the major problem. This is at the pilot scale. So that can be done if we are using any concentrator, so that if we are getting hydrolyzed in higher volumes, then we have a concentrator that can concentrate it around 60 gram per liter glucose and xylose, and that will be very beneficial. And it is a major problem in the scale-up issue. And the second major issue is the vessel that we are using at
Yeah,
our
good
lab.
enough. I think we'll see if there are more questions. We'll come back later. Thank you.
So do you have any, did you contacted any companies or commercial, did you attempt to commercialize this?
Yeah, actually, we are working with ONGC, and ONGC have given the approval for this pilot project for this plant. So we are working on that. And in addition to that, we have project of DBT also. They have also approved our pilot scale level of this our process. So on the lab scale, we have already demonstrated them all the processes, and we have got the very satisfactory results. So we
Thank
have
you.
two companies in our hand that can carry out the pilot scale process.
Hi,
Okay.
Karthik. Do you have you during this work have you started to characterize the end product, and how do you gonna do a comparative analysis for that? Because there always will be some level of productivity there, but how are you going to analyze where do you what's your cutoff to say that this is what I want?
Actually, we were aiming for the ethanol productivity of around 2.51 gram per liter per hour, but that can be achieved only in the single fermentation of glucose and xylose. In the co-fermentation, it reduced drastically because of various factors. So till now in the literature, we were able to get, I have been able to see around 1.5 gram per liter per hour, which is the highest ethanol productivity that has been achieved. So we are want to cross the threshold of that productivity. So we have achieved around 1.56 gram per liter per hour productivity, which is very good. And this productivity can be further improved by a combination of fed batch process, because we are combining this fed batch process with this co-fermentation process also. In my presentation, I couldn't show that, but if we combine that fed batch process also, then we can add more xylose after the end of this co-fermentation also. And we can add around 2 to 3 times the xylose in the end of the fermentation, and we get the ethanol from that. So we can around 5 gram per liter per hour, we can achieve our efficiency for this ethanol productivity. So it can be done.
Okay. I guess we are running out of time. So, judges, with your permission, can we move to the next candidate?
Yes. Yes, please.
Okay, great. So, our next candidate is Anushka Sen, and her poster number is BA44, and now she'll be joining us on the screen. There we go. Anushka, you can start your presentation, and you have 4 minutes to proceed. Thank you.
Good afternoon to all the dignitaries and audiences present here. I am Anushka Sen, here to present our poster entitled Genome-wide in silico analysis of oil sweet transporters indicate their involvement in abiotic and heavy metalloid stress responses in rice. Sweet stands for the sugars will eventually be exported transporters. This unique class of membrane-bound transporters not only take part in sugar transport across biological membranes, but they are also involved in diverse physiological functions in plants. Although the response of sweets in biotic stress is well documented, yet their involvement in plants' response to abiotic stress is yet poorly understood. Thus, to bridge this gap, a comprehensive in silico study of the oil sweet family was conducted to check their involvement in abiotic and heavy metal or metalloid stress. Talking about our results, initially we've identified 21 oil sweet members in rice on performing in silico studies. On analysis of the cis promoter elements of these 21 OS suites, it was found that they contain numerous abiotic and heavy metal stress responsive elements. Further, certain phytohormone responsive cis elements involved in metal stress response were also identified. This indicates the involvement of sweets in abiotic and metal stress tolerance. Next slide, please. To further validate our idea, miRNA target analysis of OS suite genes indicated that at least 8 miRNA families involved in various abiotic and hormonal stress responses have target sites in 13 OS suite members. Also, certain miRNA targets were found which show a direct correlation of cellular homeostasis of toxic metalloids such as cadmium and metals such as arsenic. On performing in silico gene expression analysis, it was seen that OS-sweet genes showed direct modulation under abiotic stress as well as under cadmium and arsenic stress. We all know that heavy metals like arsenic and cadmium can induce higher ROS generation in plants. We then anticipated that the efflux transporter suite can be involved in regulating the cellular levels of sugar, which in turn can act as cellular antioxidant and help in scavenging the ROS molecules. Next slide, please. Sorry, this is not my slide. My third slide is missing. Consistent with this hypothesis, protein interaction analysis demonstrated that the metal-ion regulated OS-sweet genes have some interaction with protein transporters which can regulate the heavy metal stress in plants. Taken together, our study clearly indicates that the involvement of OS-sweets in abiotic, especially heavy metal or metalloid stress responses in rice. However, further studies are required to analyze the proper functioning and the regulatory mechanisms of these OS-sweet members, especially the metal stress responsive members OS-sweet 12, OS-sweet 13, and OS-sweet 14, so that they can be viewed as potential candidates in future genetic engineering approaches to develop stress-resistant transgenic rice plants. Lastly, but not the least, I would like to thank Biotechnica for providing us with this wonderful opportunity of showcasing our talents on this platform. Thank you.
All right. Right on time. So, judges, questions? Judges, do you have any questions? Would you like to ask?
Not really particularly, except that did you had a chance to talk to any of the industries if they're interested in this?
Sir, actually, this is just an in silico analysis. We are undergraduate students and we just started our experiments, a wet lab work in the university. But unfortunately, it was just in the month of March when, due to the pandemic situation, our university had to be closed down. We plan to continue the wet lab research as soon as the university opens. And once we have performed the real-time PCR and further wet lab work, then we can proceed further with such kind of ideas of approaching industries.
Thank you.
Thank you, sir.
Anushka, good presentation.
Thank you, sir.
Could you please tell me, what kind of applications finally you see from this research?
So basically, as we know that in a country like India, or we can consider that in the entire world, rice is the second most important cereal. So in present days, what we see that due to different kinds of environmental stresses, say the temperature of the world is increasing at any rate, so all these generally affect the productivity of rice. And then traditional techniques are always there, but molecular biology techniques will always help improve the abiotic stress tolerance. So we have basically focused on these transporters. These are sweet, so that they can improve the abiotic stress tolerance of the rice plants, especially arsenic and cadmium. Like in India, or taking particularly the area we live, West Bengal, this is a surging problem nowadays that due to the arsenic contamination in the soil, huge populations get affected, sir. So that is why this is the approach which we want to incorporate so that it can be of benefit for the greater world. That's it, sir.
Okay, thank you.
All right.
Thank you, sir.
Thank you, judges, and thank you, Anushka. Wonderful presentation there. So now the time is up, so you can exit the backstage. And now we'll proceed to the next category. The category is rare disease awareness and management. And we would like to welcome our first presenter, Sangeeta Day, and she's there with us now. And Sangeeta, you can start your presentation now.
Good afternoon, my respected judges and all the honorable dignitaries present here. management of Niemann-Pick disease type C. Can I have the next slide, please? So first, I would like to fall light upon that of what is particularly rare diseases. So rare diseases are something that is currently affecting 3.5 to 5.9% of population all over the world. Now, our choice of rare disease, which is Niemann-Pick disease type C, that is currently affecting 1 out of 1.2 to 1.5 lakh of population all over the world. Now, Niemann-Pick disease is a particular neurodegenerative disease that belongs to the lysosomal lipid storage diseases, and that mainly happens due to the mutations in the NPC1 and NPC2 gene. And when these mutations is inherited within an individual, the individual have impaired intracellular transport of cholesterol and other glycosphingolipids. Particularly, the accumulation happens in the central nervous system and peripheral tissues. Now, due to this accumulation, the neurological and developmental problem arises within the individuals, and this mainly prevalent within the children of 4 to 7 years old. Next slide, please. Now to the next, I would like to say the etiology, that how the disease progresses. So as I have said that NPC1 and NPC2 gene, which residing at chromosome number 18 and chromosome number 14 respectively, these two gene produces a particular transport proteins that helps in the cholesterol transport within the cell. Now when these two genes are mutated, we don't have sufficient amount of proteins within our cell. So as a result, the unesterified cholesterol is accumulated within the lysosome and cell toxicity increases and cell death happens. Now, due to the cell death, we have the symptoms of our Niemann-Pick disease. So the symptoms for perinatals are mainly jaundice and cholestasis, hepatosplenomegaly, etc. In early childhood, the person may have clumsiness, seizure, dysarthria, dysphagia, cataplexy, etc. and several VSGP like vertical supranuclear gaze palsy, etc. type of cognitive impairment. And in juvenile and adult patients, psychiatric disorder and respiratory failure leads to the death of the patient. Next slide, please. Now, I would like to say the diagnosis. So, diagnosis can be done through particularly observing the symptoms. But except observing the symptoms, we can do some particular tests like plasma biomarker test, which is the presence of oxysterol and bile acid metabolites. We can also do test of NPC1 and NPC2 genes. We can sequence that. And the other test is Philippine test, Philippine staining test, which is used in ancient ages and used nowadays also. Next slide, please. So since the treatment is very important to manage a particular disease, so the treatments of NPC disease mainly includes miglustat, which generally inhibits the glycosphingolipid synthesis that deposits in our brain. There is occupational therapy like supportive care. Symptomatic treatment is also there. For swallowing problem, we can use gastrostomy tube, and we can do organ transplant also in case we need it. So in the next, I have here suggested, or not suggested, I have here mentioned some successful phase 2 and phase 3 trial drugs. So these drugs are erimoclamol, placebo, 2-hydroxypropyl beta cyclodextrin, BTS 270, N-acetylcysteine as biomarker, and also corinostat. Next slide, please. So since prevention is better than cure, so we also like to prevent the disease. Now to prevent the disease, we can use carrier detection test, genetic counseling, genetic testing of the individuals who are getting married. And the persons who have successfully diagnosed with NPC, they may not have drugs and alcohol that could increase their symptoms. And finally, though I have discussed few research avenues, but there are still research avenues are there, like gene therapy, enzyme replacement therapy, stem cell therapy, neurosteroid replacement, etc. Now, this may help in complete diagnosis and treatment of the Niemann-Pick disease type C. Thank you so much. The next slide contains my... Next slide, please. These are my references. Thank you.
All right, almost on time. So let's try to bring in our judges quickly. So judges, do you have any questions, quick questions?
No questions from me.
No questions yet? All right.
So,
So Sangeeta, thank you.
So have you studied some of the work where people have used dextrose in Niemann-Pick disease as a potential small molecule intervention?
Yes, sir.
And
I have.
work.
Yes, sir. Yes, sir. I have studied so many papers, and from that, I have collected the name of drugs like air, placebo, and how they are targeted and how they are used. So I just here entered, collected the names, and I have mentioned here. And this is sir.
Okay, thank you.
Thank you, sir.
All right, there you have it. So we had Sangeeta. Thank you, Sangeeta, for joining in. It was really nice listening to you. Okay, so now with the permission of judges, we'll move to the next participant in the same category, and she is Dr. Liji Milesh, and she'll be joining us shortly. Liji, if you can hear me,
Yes,
you can
yes,
start your
sir. Yes, sir.
presentation now.
Am I audible? Okay.
Yes, you are. Please proceed. Yes, you are audible. You can proceed.
Yeah, very good evening to one and all present here. Myself, Dr. Liji Milesh, and I'm here to present my work that is ELISA-based seroepidemiology exploration of Sura in bovines and camel in Karnataka and Rajasthan, which was completely done at ICAR Institute, that is in Vedi at Yelahanka, Bangalore. Objective of my work is to develop cell membrane-based indirect ELISA. Previous, thank you. The objective of my work is to develop indirect based ELISA, that is cell membrane based indirect ELISA for serological assay. And the second objective is to screen field sera samples from Karnataka and Rajasthan for seroprevalence study for the presence of antibodies. Coming to the introduction of my work, Trypanosoma evensi is also known as T. evensi, which is a hemoflagellate parasite and which basically causes trypanosomes or Sura, and sometimes it is also known as sleeping sickness. It is transmitted by the tablet flight and it basically affects a wide range of animals like wild animals as well as domestic animals. When the infection is too high, it can easily be detected with the help of microscopic analysis or any conventional method. But when the infection is too low, it becomes really difficult to detect Trypanosoma evensi. Thus, keeping this point in mind, we have designed a diagnostic test which will be really helpful in detecting this antibodies of T evensi, and it can be used for the mass scaling also. Next slide, please. Next slide, please. Yes. Coming to the materials and methods of my work, first, previous slide, please. Previous slide. Coming to the materials and method of my work, first I have isolated the cell membrane antigen from T evensi, that is Trypanosoma evensi, from the whole trypanosomes. Then I have characterized cell membrane antigens. Then I have developed indirect ELISA using cell membrane as the antigen. Further, I have done statewide seroepidemiological study, and then statistical analysis was done for the test. Coming to the result of my test, I isolated the isolates were revived from the liquid nitrogen. It was propagated in lab animals. It was purified using chromatography technique. Further, cell membrane antigens were isolated, immunoreactivity were checked, and indirect ELISA were synthesized. Now, in the table, I have used around 220 field sera samples, especially from bovine and camel from Karnataka and Rajasthan, and these sera samples were subjected to our newly designed indirect ELISA test. I have also used one more test as a reference test, and here the test that I have used was CAT-T evensi test, which was procured from OI reference laboratory from Belgium. So when I subjected my sera samples, that is field sera samples, in both the tests, I found out that 22.72% seropositivity was seen in my test, and similarly 22.27% positivity was seen in the reference test. Further, doing square analysis, we found out that both the tests showed very high significant value. Next slide, please. Yes. The benefit of my research outcome, my work can be used as a large-scale sample screening. It can be used for the qualitative and quantitative analysis. It shows very high sensitivity and specificity. It shows very less cross reaction, and it can also be used for the carrier sample screening. It is cost effective, it is easy to handle, and the results are consistent and good reproducibility is found. The prevalence and the mortality status can be studied as well as obtained, and it is also useful in the treatment as well as control program. Summary of my work: the present indirect ELISA design assay was observed to have very high diagnostic potentiality and is at par with the reference test. The seropositivity was found to be 22.72%. Conclusion of my study: the present study has a potentiality to be used in serodiagnosis of sura for mass screening, thus eventually will help in the detection of the carrier status of the infection. The epidemiological data available can be further used for the treatment and control strategies. Acknowledgement: the work was supported by Department
Dr. Liji,
of
your time is
Biotechnology,
up.
BBT, ICR, Nivedi, Bangalore, my guide, Dr. P.P. Sengupta sir, and I'm really thankful to the HOD of Reva University, all the co-authors, my mentor, Dr. Jairam sir, Sheva Suman sir. Thank you.
Okay. Judges, do you have
Thank
any
you.
questions quickly for Dr. Liji? Yeah, Dr. Ramesh, please go ahead.
Yeah. Somehow I missed your point on the antibody. Can you just highlight how the antibodies were obtained?
I guess her internet
Am I
is a
audible?
little slow.
Yes, we can hear her well also.
Yeah.
I think the antibody comes from the samples, the sera. The antigen only she has to say where she has the target antigens for the ELISA.
Okay. So antibody comes along with the kit.
No. The antibody is what we are looking. Serosurveillance is the antibodies are present in the serum of the suspected
Yes.
or the test animals.
Okay.
All right. So that was Dr. Liji. Unfortunately, her internet was not that great. So we had audio issues, no problems there. So we'll start with our next poster presenter and poster number RD25. And we have her on screen, Ankita. Hello, Ankita. How are you?
Hello, sir. Good evening, everybody.
Okay.
So the topic of
So
my
Ankita
poster
will
from
start.
the rare disease. Okay.
Yeah, you can start now.
Okay. Good evening, everyone. So the topic of my poster from the rare disease management category is Huntington's pathogenicity regulated by cellular translation. Can I have my first slide, please? Okay. So Huntington's disease, as we all know, is an autosomal mutation which occurs in the short arm of chromosome 4, where a pre-existing polyQ tract is again repeated. And when it is repeated beyond a number of 40 repeats, we see the onset of Huntington's disease in the individual. And this disease is characterized by symptoms like involuntary movements, hence it is called as Huntington's chorea as well, psychological abnormalities, cognitive dysfunction, and neuronal loss. Next slide, please. Can I have the next slide, please? Okay. So what we have done is we have mimicked this Huntington's mutation in the yeast background, that is S. cerevisiae. Here you see Q25 and Q103. So 103Qs are mutant. In the mutant background, we see that the growth has been reduced, the protein is aggregated, and also there is a reduction in polysome to ATS ratio. So this is an image of a serial dilution assay where we see 103Q growing slowly. The inset is the GFP tagged protein, where in 103Q we see bright fluorescent punctate indicating protein aggregation, and the reduced polysome to ATS ratio might indicate a translational defect occurring somewhere in the mutant background. Can I have the next slide, please? So this is where we've done the same thing, but we've done it in S2 cell line of Drosophila. So here also in the mutant background, that is Q138 in this case, we see aggregation in the mutant. We also see reduced polysome to ATS ratio. And another thing we observe is that we see a reduced puromycin incorporation. Now, puromycin is incorporated in translating proteins, and we see less of that incorporation, which again indicates that there is a translational defect occurring in the mutant background of the Huntington's disease. Can I have my next slide, please? Okay, so here we have studied ORB2. Now, ORB2 is a translational regulator protein, which is a prion-like protein, which is a homologue of human CPEB protein. So what we saw over here was ORB2 was getting sequestered along with our Huntington aggregates in yeast background, as well as in Drosophila optic lobe of the neurons in the optic lobe, as well as an S2 cell line. And then we did an eclosion assay where we saw that the toxicity because of the Huntington mutation was getting rescued when it was co-expressed with ORB2 isoforms. So in this graph over here, you see that the survival rate is getting increased when 138Q is expressed with ORB2A and B. And we did a similar parallel loss of function experiment where we knocked down the ORB2 and we saw that the survival rate is going down. So this indicates that ORB2 interacts with Huntington's pathways and it can modulate its toxicity. Can I have the next slide, please? Okay, so now we wanted to see the exact mechanism in which the ORB2 is affecting the Huntington's toxicity. So we tried to count the aggregates where we saw ORB2 getting sequestered with our Huntington aggregates in Drosophila neurons again, but we couldn't make out a significant difference between the number of aggregates in presence and absence of ORB2 isoforms. We also did a semi-denaturing electrophoresis experiment where we tried to compare the oligomers, but there also we couldn't see a significant difference between the neurons and the neuronal aggregates in presence and absence of ORB2. But when we did our polysome experiments and the puromycin experiments again, we saw that in presence of ORB2, the polysome to ATS ratio goes up again. And also the puromycin incorporation is increased when our mutant is expressed in presence of ORB2 isoforms. So in conclusion, I'd like to say that ORB2 rescues the Huntington toxicity, and it does so not by altering its aggregates, but by actually improving the translatory status of the cell. Can I have my next slide, please? Okay, so this is just some additional data where just like ORB2, we saw another ribosomal protein, RPL25, which is also sequestering with our Huntington aggregates. And yeah, that is all.
All right. Thank you, Ankita. So judges, let me quickly bring you on the screen. If you have any questions, quickly, you can ask.
Is there any application for this research, or you're more looking at it as a basic research?
So, sir, we've seen similar results in human cell lines also. So we've done neuroblastoma cell line where we saw the homologue of ORB2, which is CPEB, which was also getting sequestered in the Huntington aggregates. And when we co-expressed it, the puromycin level was again going up. So these results that we saw in Drosophila and yeast are also sort of applicable, or they show the similar results in human neuroblastoma, trying this in different model systems.
So quickly,
Right.
so do you have do you plan to take to small animals this study, because then you will get a much better understanding in terms of physiology, how the mutation, you have to create mutation, or the models are available. Do you plan to do that, because that will be a good job?
Yes, yes. So right now we have just done on yeast and cell lines like S2 and some neuroblastomal cell lines, but we haven't yet actually got a chance to do it in vivo in mice or anything. But we are surely planning to try that out.
All right.
Model is available. You will have to knock in yourself for it to be available in some.
Yeah, please go ahead.
Sorry, I couldn't hear you.
So one quick
I
question
will
from
quickly
my
ask,
side.
animal models are available for this?
No. I really cannot hear you. Are you asking about the animal models which are available for this kind of study?
Yes.
I think we could do this in mice as well. But I can't tell you the exact, like the strains and all that could be used.
That's okay.
We haven't done it yet.
All right. So now we have run out of time for Ankita. Thank you so much for your time, Ankita. It was a pleasure listening to you today. And now we have the next category. Judges and my dear audience, we are moving into the category 3, and that is synthetic biology. Now, before I move on to the next category, I have observed and seen that some of you are having some questions about the votes. So as you can see, 5 o'clock exactly, the votes will end. Okay. And those who have got most popular under your posters, the most popular category. Okay. So those will be getting separate prizes. That is not about presentation. For presentation, they're getting the prizes right now. They are competing against each other. But all those who are in the most popular presentation category, that will be decided as per the audience votes, which is going on the website. Okay, and there are last 10 minutes now left in the votes. So now I'd like to bring in our first participant in the synthetic biology, and that is Asha. Over to you, Asha.
Okay, thank you so much, sir. Good evening, everyone. Today I am presenting on a cybochloroplast enhancing CO2 mechanism, CO2 fixation mechanism. The major challenge for the future is increasing CO2 concentration in the atmosphere, which cause undesirable changes. One and only solution for these problems are plant. They can harness CO2 from the environment. Plant convert light into energy cofactor, ATP and NADPH. By using these cofactors, they convert CO2 into organic molecule, which eventually used by all lives on Earth. This occurs in the chloroplast in a very spatial and a temporal manner. In nature, chloroplast consists of energy converting module and fixed CO2 and CO2 fixing module. Can I have a next slide, please? Yeah, so usually we look at CO2 being a problem, but at the same time, we can take it as a chance to make useful compound out of it. So if we think about the plants, they are efficient CO2 fixing filters, but they are not fast. Motivation behind this research is to find a new way to fix CO2, and there's always chance to improve existing biology with synthetic biology. The problem with the existing biology is CO2 fixation is the main enzyme Rubisco, which is very slow catalyst. In 2016, the Swender and his co-worker found another class of enzyme called ECR, enoyl-CoA carboxylase reductase, which can fix the CO2 10 times faster than the Rubisco. Idea was to reinvent CO2 fixation on the basis of this enzyme, which discovered by metabolic retrosynthesis. At the end, it has been 17 enzymes from the 9 different organisms and some redesigned. But the challenge was to bring them together and harmonize to work together as a team. And finally, they got so-called catch cycle that can catch CO2 efficiently from the environment. As we know, plant has efficient energy converting module. On the other hand, we have the sketch in a lab is efficient CO2 fixing module. Now the next challenge is to bring these two modules together. For this, we can use microfluidic to prepare oil water emulsion droplets, which act as an individual cell. In single droplet, we can co-encapsulate both the energy module, that is thylakoid membrane taken from the plant, and the CO2 fixing module, that is a catch cycle. When these droplets illuminated, the thylakoid membrane are able to generate ATP and NADPH as it generates in the natural system, which can be utilized by the catch cycle for the continuous CO2 fixation into organic molecule. The major challenge for the scientists is whether these all things going to work together and all. Finally, they got success and it actually worked. I have the next slide? Can I have the next slide? So now the question is, we have to bridge a gap between the biology and the chemistry. So how can we do that? It has the 5 steps. Either we can manipulate, modulate, and knock out the existing pathway, and we can copy the enzymes. We can copy the pathways with the enzyme into the plant. And now the next one is what we can do. We can just with the known enzyme, with the known reaction. And in the fourth step, what we can do, we can in the cycle, we can engineer the enzymes according to our need, and we can introduce in the plant. And in the last one, what we can do, we can create our own novel enzyme with a novel cycle, and we can introduce in the plant. Now, what is the benefit of this study is like that in this, what we can do, we can transform the greenhouse gases into the fuel and food for the growing population. And these reactors can be solar powered. These are autonomous reactor. They can create their own energy to work for themselves, and they can be regulated from outside by the light. These microreactors can be studied side by side in a real time, as they are independent from one another. This energy independent synthetic cell can be used for the drug production and artificial metabolic cycle at a nanoscale in a high throughput manner. Now what we can do, we can edit and improvise the natural pathway and can create new to nature to improve our nature, which is already there. But it all has come at a cost. We should not forget the consequences. Difficult to actually, this catch cycle, it's really difficult to introduce back to the plant, because this catch cycle has an enzyme which is derived from the 17 these 17 enzymes derived from the 9 different organisms, which has a different evolutionary history, and it is very hard to work as a team. Now next slide please. Here I'm concluding my seminar. I just want to convey a message, like try and leave this place, this world a little better than you found it, as in the word of Robert Baden-Powell. Thank you so much. I want to thank Mayank for helping me for this. He's my co-author. Thank you.
Okay. Thank you, Asha, for your wonderful presentation. Bringing in the judges now. Judges, if you have any questions, you can ask Asha. Over to you, judges.
This is Shishida. You said it was difficult to put back into plants and something new to nature. I was wondering if this technology could be useful to develop plants that can be cultivated not on Earth, maybe on the International Space Station, or maybe in future on the Moon or the Mars. Because we are talking about completely new system that is needed there. So you could consider working with ISRO or other organizations.
Yeah, sure, sir. It's really interesting. What I was thinking, sir, by using these pathway, what we can do, we can prepare some small, the tubes of the, like having glucose and all, and we can just take it to the space. That microfluidic should have the enzyme which can convert all this, and it should be the it should give energy by itself, like as plant is giving to themselves. So they can create a food there, and they can like have it and do research there.
Actually, space in International Space Station, that's physical space, experimental space, is available.
Yeah.
And you can lease it. You can work with NASA or with JASA or these organizations. Actually, we have designed some experiments on the ISS, so you can you can work with ISRO and many scientists. It's a very interesting concept for not necessarily Earth. Something beyond. Thank you.
Thank you so much, sir.
All right. Thank you, Asha. Judges, if you have any more questions, you can ask now. Otherwise, we'll proceed to the next participant. No questions? Okay. So thank you, Asha. That was a wonderful presentation. You may leave the backstage. All right.
Yeah. Thank you.
So our next presenter in the same category is Rashni Shetty, and I'll quickly bring her on screen. Rashni, over to you.
Good evening. I am Roshni, and I wish to give a brief overview of synthetic biology and reprogramming cells. What is synthetic biology? Today, we have the tools to cut and paste DNA, allowing us to manipulate different genes using scissors and glue to give us unique combinations and functionalities. It is just like a car. It can be customized. The process of choosing what to add or remove, finding the spare parts, and assembling it back is the science side of SynBio. Next slide, please. Imagine designing an organism by assembling a jigsaw puzzle with the functions we desire. Can we produce anything and everything? For example, scientists were able to copy and paste the gene that makes fireflies glow into zebrafish and make it glow too. This appears useless, but what if it glows only when the water is polluted? It becomes useful. These puzzle pieces or distinct functional units can be defined as a module. We currently have many standardized biological parts or biobricks promoted by the iGEM competition, which aims to encourage students and researchers around the world to contribute to the biobricks repository. Examples of biobrick parts include promoters and terminators. Next slide, please. Will it always work? We can imagine a biological system as analogous to an electrical circuit. Upon the connection of different parts, impedance-like effects. In biological terms, retroactivity emerges. We must always remember that the whole is not just more, but also very different than the sum of its parts, and what may work in one system may not work in another system. Molecular crosstalk and feedback signals always exist. Biologists, if not frustrated, are always humbled by the inherent complexity of living systems. Synthetic biology has inspired by electronics and software design. Successful amplified biosensing, time genetic circuits, oscillators, and switches have been designed. As Jay Kiesling said, with the tools of synthetic biology, we don't have to just accept what nature has given us. A renowned example is the synthetic production of anti-malarial drug artemisinin for falciparum malaria. Traditionally, artemisinin is obtained from the sweet wormwood tree. Now we can produce it by the fermentation of genetically engineered yeast, which is much more efficient than using the plant and has reduced the environmental impact. Next slide, please. Today, so many substances are being produced synthetically. Insulin, growth hormones, clotting factors, and vaccines. Many substances in agriculture, such as fertilizers and PHP plastic polymer. Microorganisms can absorb oils, detect toxic metals, and are used in waste treatments. Inserting genes into plants to confer antimicrobial resistance and increase their shelf life. The enormous applications in the diverse fields are numerous and breathtaking. But one also has to consider the safety side of genetic engineering. Consequences of modification should be clearly predicted and analyzed. Biosafety and ethical concerns are the need of the hour. We are for sure that synthetic biology will revolutionize our world. Its vast potential and promise lies in understanding the comprehensive processes by reflecting on the complexity and specificity. Thank you. Next slide, please.
All right, so there you have it. Roshni, wonderful presentation, really wonderful. Let me quickly bring in our judges on the screen and we can start with the questions now. Over to you, judges.
No question from my side.
Right. Roshni, can you tell me what do you think are going to be environmental impact of synthetic biology if you're going to go on changing the wild type varieties?
Currently, we can modify plants, say to increase, have an antibacterial resistance and even increase their shelf life. But when we even engineer organisms, we should be careful on the environmental impact it has. So the GMOs, in the long run, what if we know that microorganisms are co-evolving and living, even though they might be parasiting and harmful for one particular species, but it might be benefiting another species. So if you completely engineer an organism to eliminate one species, might be harmful. So in an environmental perspective, GMOs can be bad.
Okay, thank you.
All right, any more questions, judges?
Thank you.
Okay, looks like Roshni, you had a powerful presentation there. Thank you so much. You may leave the backstage now. And now we will bring in our next participant. Okay, and in the same category, Falak C. Pandit. Over to you, Falak. You can start your presentation.
Am I audible? Hello?
Yes, you are audible. Please proceed.
Okay. Good afternoon to one and all present here. My today's topic is synthetic genome, rewriting the code of life. First of all, what is synthetic genome? Genome implies to the overall genetic material of an organism, and synthetic means artificially synthesized. This means either the whole part of genome or a particular part of the genome can be synthetically modified. Now, I would like to mention here the two major milestones in this field of the synthetic genome. That is the creation of the first cell with the synthetic genome and the first synthetic eukaryotic organism. Now, the first cell with the synthetic genome was the M. mycoids in 2010, and the other was the Saccharomyces cerevisiae yeast, that is the eukaryotic organism, in 2014. The pictures of both the organisms can be shown on the slide. Now, I should come to the question, why synthetic genome? Now, if we design a particular set of genes and we remove the excess of the genes from an organism, we can design a metabolic pathway into such a manner that we can get the desired products. So this gives idea to the minimal genome concept. Now, what is minimal genome concept? Minimal genome concept means a particular set of genes or a minimal set of genes which are required for an organism to survive and proliferate in a given condition gives rise to the minimal genome concept. Next slide, please. Okay, so how the synthetic genome is actually organized or synthesized? There are 5 steps: concept, design, modeling, simulation, and validation. To go on a easier pathway, I would like to mention if we take a genome of a model organism or a particular organism, we get the whole genome sequencing data or a part of the genome sequencing data which we want to synthesize artificially. Nextly, what we do is synthesize a particular DNA fragments. The smaller fragments, then we assemble into the bigger fragments. So if you can see in the figure given on the right hand side, we can see the smallest fragments of the DNA have been synthesized, then they are assembled into the bigger fragments, and then the whole genome assembly. Next is the DNA transplantation into the high capacity cloning vector, and then screening to see whether our organism has actually took up the synthetic genome or not, or either it is expressing its own genome or the homologous recombination has occurred or not. Last slide, please. Next slide. Okay, so what are the applications of the synthetic genome? It can be used in plant genomics, therapeutics, and genetic engineering. Now, plant genomics, we can create certain genetically modified organisms, and we can produce the biosynthetic molecules such as artemisinin and Taxol that can be used in the diseases such as malaria. Nextly, we can synthesize the synthetic circuits. Synthetic circuits may be consist of the promoter, activator, and the gene, and these prosthetic devices can be further used for the treatment of the certain diseases. Another application of it implies in therapeutics, where we can modify particular metabolic pathways of particular diseases, and we can there reach to the treatment, such as the gene therapy can also be used. Otherwise, industrially, we can genetically modify a particular microorganism, and we get the desired products through the industry. Last slide. Thank you so much. I would like to acknowledge Biotechnica for providing such a great platform, and thank you so much.
Okay, thank you, Falak. That was really insightful and great presentation indeed. Judges, if you have any questions, you may ask now.
Yeah, did you ever do any experiments? Did you do on the in silico or on the lab? Looks like you just reviewed something and put it.
So practically, I have not done any such. I have completed a dissertation project, but it was not based upon this. It was based upon some different topic. But I have been studying a lot in this area, synthetic biology.
Thank you.
Falak,
Yeah.
you were telling about genes, useless genes or something in the last but one slide.
Yes.
Yes.
Minimal
Actually,
genome
these
concept.
Actually, these are the sites which control epigenetic behavior of any organism. These are not structural genes. They don't produce mRNA or not directly associated with any action in inverted commas. But epigenetically, they have a lot of activity. What do you say about that?
Yes, sir. If we go into the minimal genome concept, there are actually of the NOGD, that is non-orthologous gene displacement, and COG, that is conserved ortholog genes. Now these two mainly combine and form a minimal genome concept. That is, these genes cannot be removed from a microorganism, otherwise it would not be able to survive. Even though some of the microorganisms they have synthesized, like if we see say JCBI 3.0, the first organism had around 900 genes, and the third series of the organism had around 473 genes, out of which 149 are still uncharacterized genes. So we cannot actually know which genes are playing role in the epigenetic evolution. We have to still characterize the function of each and every gene. Then only we can design the minimal genome concept pathway. The work is still going on in this area.
All right. I hope there are no more questions for Falak. All right. Thank you, Falak, for your time. It was wonderful. All right. So, judges, quickly starting with the next category, which is the fourth category of today, that is Omics. Omics is an upcoming field. It's a very interesting field. And today, with us is our first participant, that is Ketki. Ketki Gatole will be joining us now. Yes, hi Ketki, how are you?
Good evening, sir. I'm fine.
Yeah, so you can start your presentation now.
Yes, sir. Yes, sir. So, good evening, everyone. This project mainly focuses on understanding the evolution of venoms through codon usage bias and amino acid preferences. So, venoms are pharmacologically active complexes that include proteins, enzymes, and various non-protein compounds. As venoms have specific effects on various biological functions like blood coagulation, blood pressure regulation, they have a wide range of applications in the pharmaceutical industry. So, most of the research in the field of venoms is mostly focused on its clinical implications rather than actually understanding the biology or evolution of venoms. Research in venoms is extremely difficult because it's available in highly complex mixtures and in very limited quantities. However, with the development of omic technologies, it's now easier for the researchers to identify the genes that produce these venoms and the proteins that are building blocks across these venoms. So, understanding the variation in the gene expression and the coding sequences can help us understand the variation in venoms across ecological and evolutionary timescales. So, understanding codon usage bias can give us an understanding about its phylogenetic evolution. Different factors such as amino acid frequencies, RSU, which is relative synonymous codon usage, GC content, and effective number of codons, that's ENC, play an important role in shaping the codon usage bias. The main objective of this project is to identify the evolutionary pressures and constraints that shape the landscape of natural venoms. Next slide. So this diagram gives an overview of our experimental workflow. So the amino acid sequences of venomous proteins for the taxas Araniae, Chilopoda, Nidaria, etc. were obtained from the database Venom Zone, and the amino acid sequences for the non-venomous proteins were obtained from Uniprot. And the coding sequences for all these proteins was obtained through NCBI, and the data was analyzed in DAMBay, that is data analysis in molecular biology and evolution, to obtain the RSU values, GC content, ENC values, dinucleotide frequencies. The results were further analyzed and visualized in Microsoft Excel, Python, and CIM Miner. So the results obtained were as follows. The first study was the RSU analysis, and the picture on the screen is the heat map or the clustered image map, where a darker color indicates higher frequency. So the codons were divided into two categories based on AU ending and GC ending, and the left side where the darker color is seen is the one with AU ending. So this shows that the venomous proteins have a higher preference for AU ending codons. Next slide. So the next study was amino acid preferences. It was seen that venomous proteins had higher preference for cysteine as compared to non-venomous proteins within each taxa. And the Euclidean distances gave an insight about the genetic distance between the selected taxas. And within each taxa, it was seen that the venomous proteins and non-venomous proteins had had different preferences for amino acids, showing that they have evolved independently. The next study was the GC content. So it was seen that the GC content was lesser in the venomous proteins, and it was approximately around 40%, showing that venoms prefer AU ending codons. And the next study was ENC versus GC3. So in this study, it was seen that only 25 out of 4,200 coding sequences had a strong codon bias, and it suggested that mutational pressure had very little role to play. The next study, that's neutrality and parity plot, also gave similar results, and it was seen that only 26.19% of the total effects were because of the mutational pressure, and other factors like translational pressure had more role in playing in shaping the codon usage. So we can conclude by saying that venoms have evolved independently at different time scales in different taxas, and this evolution has been extremely complex and beyond the phylogenetic constraint. And venomous and non-venomous proteins within each taxa has evolved independently, and venomous proteins prefer AU ending codons, and mutational pressure had very little role to play as compared to other factors. So these conclusions can help us build a genetic profile for these venomous proteins across these taxas. So this data can further be used and incorporated in different machine learning approaches that can be used to identify and classify these venoms, which can further help in developing peptide-based drugs in the pharmaceutical industry. Thank you.
Okay, thank you, Ketki. That was really insightful, exhaustive study. Quickly, we'll try to bring in the judges here. Judges, if you can come in. Yeah, over to you.
I'd like to get some comments from Ketaki on a different perspective. Suppose if you were to take the proteins that are involved in venom formation or the venoms themselves and compare across the groups whatever is homologous protein or the genes for them, the coding regions, and then do whatever analysis you did, what type what type of advantage or disadvantage you would have seen in the strategy?
So the thing is, the reason we selected these proteins was because there were very few databases that actually gave us like protein sequences or coding sequences for venoms. So we selected one database and we took all the data that we could get from the database for this analysis. So the main reason we selected was because of the availability.
If you get a protein sequence, you can find whatever is homologous. You can get the sequences for that. You can that, right? You don't have.
Sir.
So if you had taken that approach, what would be the advantage and disadvantage?
So actually we wanted to do this study like to just compare the amino acid sequences. So homologous studies we could have done, but like we wanted to study more on a molecular level and understand the preferences.
Okay, quick question.
Yes.
What about the rest of the proteins in the same snake? Does it also look like this or it's different?
So we took proteins from like we selected the 6 categories which were all venomous, like all the organisms that produced venoms. So we took all the venom proteins and all the non-venom proteins in each of those organisms. So we had a lot of proteins, like all together, including the venomous and non-venomous proteins, we had approximately 7 million different sequences. So we have analyzed that as well, and we've compared within each taxa the venomous and non-venomous proteins.
Okay.
All right. Thank you, Ketki. We have a question. Dr. Shashidhar, please come in.
Yeah. What would happen if you include the toxin from the poisonous mushrooms? Did you?
Sir, we did not really focus on, we just focused on the animals. So we didn't really take the other organisms or other mushrooms or plants or anything. So maybe we can do that. We'll consider it. We'll try incorporating it, sir.
Okay. So I hope that answers all the questions. Judges, with your permission, can we proceed to the next participant? Yes? Okay. So thank you, Ketki. You may leave the backstage now, and we'll proceed to our next participant.
Yes.
Let me
Thank you,
quickly
sir. Thank you so much for this opportunity.
Thank you. So the next on the list is Mr. Varun, and he joins us now. Hello, Varun. How are you?
Fine, sir. Thank you for the opportunity. Yeah.
So you can start your presentation now.
Good evening. My name is Varun, and my co-author is Raina. We're final year students of RV College of Engineering, Bangalore, and our poster is titled "Transcriptomic Analysis of the Entorhinal Regions of Symptomatic and Asymptomatic Alzheimer's Patients." Asymptomatic AD is a condition where patients have intact cognition but neuropathology similar to AD. Asymptomatic AD patients are highly likely to develop AD as well. So we can think of asymptomatic AD as an intermediate disease state between normal condition and AD. The intuition behind this project is that understanding the molecular mechanisms that govern asymptomatic AD could help us in early detection and treatment of a patient before AD symptoms develop. So in the methodology, we first took gene expression count data and some pre-processing such as removing batch effects and bad quality probes were done. The data are split into 3 study conditions: AD versus asymptomatic AD as well as control, and asymptomatic AD versus control. We then performed a differential gene expression analysis using statistical tools and identified the significant genes. We simultaneously performed co-expression studies as well. Then overrepresentation analysis and gene set enrichment analysis were done to identify the significant pathways and the gene sets. Finally, protein interaction networks were constructed using the co-expressed genes. Next slide, please. Now let us look at some important results from the study. The first slide is for the AD versus asymptomatic AD and AD versus control cases. We observe in the volcano plots of these 2 cases that most of the genes are overexpressed in the disease condition. Then a table of the top 5 differentially expressed genes is also shown with the p-values. We obtained thousands of genes of differentially expressed genes in the AD condition with p-value less than 0.05. In the protein interaction networks, each module represents genes with similar expression profiles or similar co-expression. We can see that proteins of genes such as SNAP25 and FOS are highly pronounced as hubs in these networks. And these are the classical genes of related to AD, thus validating our results. Next slide. Okay, so here we have the result for asymptomatic AD versus control. Most genes are overexpressed again as compared to healthy individuals, but not as much as the AD conditions. We only obtained about 40 differentially expressed genes here, and the top 5 are given in the table. Interestingly, we obtained 5 different modules for the asymptomatic AD case, suggesting that multiple gene sets are involved. In module 4, you will notice APOE4, a gene synonymous with AD as a hub of the network. So studying the other genes involved in the same module could explain how APOE4 actually acts. Next slide. Next slide, okay. The Venn diagram of the number of differentially expressed genes is given in the left side. An interesting observation is that there's no single unique gene involved in asymptomatic AD. Also, about 5,000 genes are common between AD versus control and AD versus asymptomatic AD condition. To the right, you will see the Venn diagram of the significant pathways we observed, and it also presents an intriguing result. We observed that about 17 pathways are unique to asymptomatic AD condition itself. The table to the bottom left shows the classical perturbed pathways known through literature and observed in the study. The table to the right shows the pathways in asymptomatic AD. We discovered several interesting of such pathways to be perturbed. Innate immunity pathways and neutrophil degranulation were shown to be affected, which indicates that there is an immunological aspect to asymptomatic AD onset. Coming to the conclusions, we can say that our study was able to capture pre-existing knowledge about genes and pathways involved in AD progression and also uncover new mechanisms. Finally, we proposed 3 new targets for Alzheimer's by inspecting the results obtained, and these can be confirmed by wet lab studies using gene knockouts or gene editing. So first, we observed that SIT1 was an interaction hub in the protein interaction network of all the cases. Further analysis showed that SIT1 is involved in a pathway that leads to A-beta plaque generation, which is an important biomarker of Alzheimer's. Secondly, we observed that SDC4 is a co-expression hub in all the cases. By doing similar analysis, we discovered that this gene is also involved in A-beta plaque generation. Finally, we observed CYBB as the most differentially expressed gene in the asymptomatic AD condition. This gene is known to be involved in multiple sclerosis, but its function in AD is still being researched. So we would like to conclude by saying that the results mentioned in this poster depicts only about 20% of the total work we have done. A similar analysis was done for the temporal, frontal, and cerebellum regions of the brain as well, along with additional analysis. Thank you, everyone.
All right, thank you, Varun. Quickly bringing in the judges now. Judges, if you have any questions, you can ask now.
So, Varun, this was a public data you used.
Yeah.
So how different was your analysis compared to the original analysis, number one? And what was the sample size and what is the type of microarray platform?
So it was very different. They mainly focused on differential gene expression in the first case. So we extended the analysis to co-expression studies and building protein interaction networks as well. And also you can see, and we also try to find the most perturbed pathways in the study as well.
What was the sample size?
For asymptomatic AD, it was about 40, and for AD, it was more, so 50, and control, it was about 30.
What was the microarray platform used?
I'm not so sure about that. Yes, sir.
Yeah, Dr. Ramesh, please go ahead. Your mic is muted. You need to unmute it.
Hello.
Yeah.
Yes, sir.
Yeah. How specific and significant is the gene which showed expression and you are considering as a biomarker?
So we got a p-value of in the power of e raised to minus 20, so which means that it's very significant. And we only considered in the table given, we only considered top 5, and all of them have very less p-values. Again, p-value is an indicator of significance. So, and we took a p-value of less than 0.05 for significance as well. So yeah, most of these genes are, again, we validated these results with literature as well. So a lot of genes such as APOE4 and SNAP25 and FOS were ever present in our study and in the networks we constructed.
Yeah. Any more questions, judges?
No questions, but good comment. Good presentation, Varun. Well done.
And thank you to Biotechnica for giving us this opportunity. It was really nice.
Most welcome. Okay. Thank you, Varun. You can leave the backstage now.
Yeah.
And judges, now we move on to category number 5. Category number 5 is stem cell and regenerative medicine. Very important category from life science perspective. But before we actually move on to this category, I can see some questions coming in into the chat box of YouTube, citing some confusion about the number of votes and why they were not allowed to present. So the most popular presentation or the poster is something different category. Okay, the live presentation is a different category. Okay, so the people who have got highest votes, they are most popular posters, but they will not be given a chance to present live. Only the posters which were selected by our judges and jury will be allowed to present live. Okay, I hope that makes it clear. There's no bias and there's no such thing as we're trying to side with anyone. It's totally neutral. Okay. Now, with that, having said that, we would like to bring in our first participant in this category, Chandra Ratan. So Chandra Ratan has joined the backstage. Yes. Hello, Chandra Ratan. Can you unmute your mic, please? Yeah.
Yes, sir.
Yeah.
Hello, sir. Good morning. Good evening to all. Am I audible?
Yes, you are audible. Please start. There seems to be an audio issue with you, Chandra. Yeah, please go ahead.
Good evening to all Biotechnica team and judges and viewers. My name is Chandra Ratan. I'm from Gautam Buddha University. Currently, I'm pursuing integrated MTech Biotech. In stem cell category, my poster tells about the beta cell isolate-like organoids which are derived from induced pluripotent stem cells. These are, next slide please. As we know, in India, diabetes cases increases every year. 1 million cases every year of rises in India. So to overcome this problem, we develop a human isolate-like organoids. These are very similar like to beta cells. Beta cells and these organoids derived from induced pluripotent stem cells. These pluripotent cells these derived from adult human stem cells, and this is done by Shinya Yamanaka, a Japanese researcher, in 2006, which he took up these four factors: OCT4, SOX2, and KIF4 CMYC. These defined four factors give to these generate a induced pluripotent stem cell. Then we differentiate into the human isolate-like organ, the beta cells. Beta cells in diabetes, beta cell destruction. To overcome this problem, that's why we develop these organoids. In these organoids, a estrogen-related receptor, they are expressed on the surface, which help to metabolically maturation of beta cells, which are help to stimulate the insulin secretion, and according to the blood glucose sugar level. And second is the wind force signaling, and non-canonical wind force signaling it is derived from wind signaling, and it also help to maturation of betas, stimulate the insulation of secretion according to the blood glucose level. Next slide, please. So the main thing and the important is the program death ligand 1 expression. This receptor expression is in the beta cells to increase this expression for the immune evade. What happen in diabetes? In patient need to take a immunosuppressive drugs. Those immunosuppressive drugs are very harmful, and some gave to, but this program death ligand expression is to overcome this problem, to induce this expression induced by the interferon gamma pulse. If we gave the interferon gamma, then you can see it there, the right side upper, the expression of program death ligand when increase in the receptor. And the second is the thing is that we don't need in this therapy, we don't need to give the patient insulin, and don't need to give the device independent therapies, and also not give the immunosuppressive drugs, which are very harmful, very effective, and it is also long-term therapy. And it is only for those people who are in type 1 and late stage type 2 diabetes. In the right side, the photos shows the human isolate-like organoids. These are very similar to human isolates. Those photos are shown. They are very similar, and beta cells are shown in granules shown in beta cells and alpha cell. Beta cell for insulin and alpha cell for glucagon to maintain the blood glucose level. But the difference between the human isolate-like organoid and cadaveric transplant. Cadaveric transplant, it gives immunological response. To overcome this problem, we derived these isolate-like organoids to overcome this problem. Next slide, please. The main conclusion is the study we need to more precise study this program death ligand 1 in animal model, in mice model, and other human models, not in primates, and also in last stage in humans, and it gives promising strategy to immunocompetence. Thank you.
Chandratan, your time's up. Your time's up. So judges, you can ask the questions now. Over to you, judges.
Yeah. So I couldn't hear in between, Mr. Chandra. Could you tell if you did cultures, what was the type of culture conditions used?
No, did you do any experiment? Looks like it's.
Yeah, I was lost in between. That's why I wanted to know if you did any experiment. Did you do culture?
Sir, I got this from a website, from Nature website.
Okay.
I gave a reference.
Good.
Sir,
okay, thank you.
these are the 3D culture medium, a gel medium in this.
Use in that paper. Okay, thank you.
Yes.
Okay, I hope there are no more questions, judges. All right. So, my dear audience and judges, that was Chandra Ratan. And now we will move on to the next participant quickly. Now, our next participant seems to be having a small technical glitch. His name is Anand. So we will move on to our next participant after that. And that is Nilofer. Okay. Nilofer, can you hear me?
Yes, sir.
Okay. So Nilofer, quickly, I'll display your presentation. Hold on. Okay. You can start your presentation.
A very good evening to all of you, respected Shekhar Suman sir, CEO Biotechnica, eminent panelist, my poster mentor, Dr. Sudha Warrior, my guide, Dr. Prakash Paligar, Dr. Manoj Garg, and all the dignitaries present. I'm Nilofer Kareem, and I'll be presenting on human-induced pluripotent stem cells, an innovative approach for the treatment of alpha-1 antitrypsin deficiency. Next slide, please. So to start with, what is an alpha-1 antitrypsin deficiency about? As you can see, in a normal healthy body, the hepatic cells in the liver make a wild type A180 protein, which is a 52 kilodalton glycoprotein. When it is made so, it is released into the bloodstream, wherein it acts upon neutrophil elastase. Now, on the degradation, neutrophil elastase is no longer available and cannot go to lungs to degrade it, and hence we have a healthy lung and liver. Now, moving on to the second case, which is a disease state, the hepatic cells undergo a single base pair mutation wherein a glutamate residue is replaced by the lysine residue at 342 position. This results into the formation of a mutated A180. Now, this mutated A180, firstly, is not secreted into the bloodstream. Secondly, is retained inside the liver. And third, cause of all these events, globules are formed inside the liver cell, leading to hepatic stress. Also, as neutrophil elastase is not degraded, it reaches the lungs, degrades it, and causes a condition known as emphysema. So question rises, how do we treat this? We have taken human fibroblast cells, made human iPSCs out of it. Next, we will be making hepatic-like cells from it. And these hepatic-like cells will be made to secrete enhanced levels of A180. Now, when such a cell will be engrafted in a diseased liver, as you can see, it will decrease the existing hepatic stress in the liver. Also, it will add to the longevity of the patient and will also help ease emphysema. And this is where our work comes into count. Next slide, please. So as you can see, many techniques and therapies have been into use, like organ transplantation or the augmentation therapies, but all these have their own limitations and barriers. Whereas stem cell therapy up till now hasn't been used to such an extent to treat A180 deficiency, which we are planning and have done a part of work to do so. So with the help of our technique, we are not only reducing the existing hepatic stress, but will also be easing the emphysema existing. Next slide, please. So this is a schematic work done by me. I've taken an Excel Blue strain, which was first of all made competent. And this Excel Blue strain was then transformed with 3 plasmids: STEMCA, which contains pluripotent genes, OCT4, SOX2, MYC, and KLF. Then PSPA-X2 and PMD2G, responsible for virus packaging and envelope. After a few hours, the DNA was again isolated and transfected into 290 3T cells. Now again, the virus particle was extracted containing the pluripotent genes, and it was used to transduce human fibroblast cells. Later, after a few days, we could see the formation of a compact clump of cell to be called as iPSC. Now, this was confirmed by following 2 tests up till now, that is alkaline phosphatase staining and Western blot for OCT4 and SOX2. A few more tests needs to be done. So we'll be further making hepatic-like cells out of it. Also, we'll be further proceeding with 3 types of cells. Wild-type hepatic cells expressing normal levels of A180 to be taken as positive control.
are generated hepatic-like cells, which are expressing enhanced levels of A180 as the test cells, and we would be generating mutant hepatic cells expressing lower levels of A180 to be taken as negative control. And so we would be seeing how efficiently we would be able to treat this disease. Thank you.
All right. Thank you, Nilofer. Wonderful presentation. Bringing in our judges. You were dot on time, Nilofer. So, judges, over to you.
Yeah, actually, it's really good work, but I think you have brought down your good work by copying images from Google. You could have, and there is nothing like Google. Google is actually showing other websites, right? So you could have drawn all those images nicely along with your co-workers or yourself. It's not, it's just cartoons. Otherwise, the work is good.
Yeah, I'm sorry to interrupt, sir, but to be very clear, the scientific poster that I had submitted in this particular competition included all my scientific results only. But later we were conveyed that we need to be more communicable to the audience so that they can understand what the work is about. So that's why I reduced my result work slides. That's all, sir, nothing else.
All right. So, if you were to continue this work, what would be the next thing you would like to do?
Sir, I would like to clear any further upcoming hurdles and would like to take it to human trials and would see how efficient it is.
Human trials?
No, sir. I said after clearing the incoming hurdles and the barriers in between. And then I would like to proceed up to that level which I have in my mind, sir. But it is a long way to go, I feel, because how efficient are will be my HLCs, and would they be secreting that great amount of A180 so that it helps cure the disease? That remains to be a main thing.
All right. Any more questions, judges? I presume there's. Okay, Dr. Satya, please go ahead.
No. Again, no question. Good presentation, Nilofer.
Thank you, sir.
That was indeed a great presentation. Thank you, Nilofer. You may leave the backstage now. And judges, next, we'll move on to the next category, which is antimicrobial resistance, category number 6. It has been a really wonderful presentation so far by many of you. But now that we have Khyati Sharma, our first presenter of the day, and she'll be presenting her first presentation. Over to you, Khyati. 1 minute, I'll just move on your slides. Yeah, that's Khyati. Over to you, Khyati.
Hello, sir.
Please be loud.
My name is Khyati Sharma, and I am presenting in my poster topic is antimicrobial activity of chia seeds, pomegranate seed, and sunflower seed with husk and without husk. These seeds are locally available in the market. So as the nutritional content and the chemical composition that people should be aware about this thing in the hustle life. There are people not completely used as a nutrition concept, but these seeds we can munch easily, and even powder form we can use in a home level based, but even industrial based to also used. This is the purpose to find the antimicrobial activity of the seeds. In this analysis, I had used 3 different polarity solvent, acetone, ethyl acetate, and methanol. By using a well diffusion method against 4 different pathogenic strains, Bacillus cereus, Asorius, E. coli, and Enterobacter aeruginosus. I can it is antibacterial activity. It is simply I explain that it is only to suppress and destroy the growth of microorganism by any tool of technique, that is the chemicals, heat method, and antibiotic drug. Seeds are as a medicine widely used since in ancient India. The phytochemicals contain terpenoids, alkaloids, and polyphenols are present in a qualitative taste, that is indication of the inhibition of microorganism. The purpose of the study to find this antimicrobial activity of the seeds. Next, please. The material I had procured from Anand local market, and the seeds are used as a powder form in 20 gram of powder and 50 ml of solvent used, which is 80% aqueous, by extracting this sample. For after extracting this sample using a incubate in a 4 degree centigrade temperature in stored in vials, and well diffusion method using with the help of a 5 bore, and the result analyzed the radius of the zone of inhibition. In the result clearly said that pomegranate seeds have a very good content in zone of inhibitions observed, and very good quality of suppress the growth of bacteria, which is commonly present in our harmful in a human body. For and the second and on other side, the sunflower seed without husk, there is no growth of inhibition observed in E. coli and Enterobacter aerogenesis. The Enterobacter aerogenesis, the negative gram of bacteria is only give zone of inhibition in pomegranate seeds, while the Bacillus cereus, Staphylococcus aureus, and E. coli give the zone of inhibitions in the another all the seeds. The methanolic extractions give a good result comparative to among the extractions. Next, please. So after observing the result, I conclude that the content of the potential of the seeds, antimicrobial activity and antioxidant properties that can be used to develop functional food for produced and herbal preparation as natural agents containing antioxidant and antimicrobial activity. For the future and present prospect.
Okay, looks like you ran out of time, Kathy. The time's up. Judges, if you have any questions, quickly, we can have you in. Yeah.
So maybe a quick question from my side, again the same, like what would you do, what would you like to do immediately after this if you are allowed to continue research?
I would like to prepare those kind of products to incorporate these seeds and give good nutritional and functional food products.
No, so specifically what would you like to do immediately next? That's my question. Eventually, let's forget that.
I would like to search another seeds also. I would like to another seeds also to search the activity of those kind of helpful in another disease and useful things.
Okay, thank you.
So, do you think this kind of a functional food or a food as medicine, is there an application for your work?
Yes, definitely, sir. I believe that the kitchen is a laboratory and food is the medicine. That's the thought, I believe. And actually, this is the way of the ancient India are also in treating the patient in the same way.
Okay, thank you.
Thank you, sir.
Thank you, Kathy. That was really insightful. Now, judges, with your permission, I would like to bring in one candidate. He could not join previously. His name is Anand, and he's from the stem cell category. I would like to bring him in now so that he can also present. Anand, over to you. Hello, Anand. Can you hear me?
Slides. Yes, sir. Slides, please.
Go ahead.
Good evening, everyone. Good evening, everyone. We'd like to share our experience of evaluation of outcomes in case of hematopoietic stem cell transplant with an flow cytometry cross-match approach to see the effects of donor-specific anti-HLE antibodies. Next slide. So, hello.
Yes, go ahead.
Can I get the next slide, please? Hello. Hello. Sir, the previous slide, please. Yeah, thank you. So the allogenic hematopoietic stem cell transplantation of CD34 positive cells holds gold standard for various hematological disorders, and this is very sophisticated treatment which requires keen selection of the donor and harvesting from them and infusing to the patient and taking care comprehensive care of the patient during and after the engraftment phase. Next slide. HLA or human leukocyte antigen is a group of closely linked gene which is present on the short arm of chromosome number 6, and it has a role in immunity purpose and organ transplantation. The outcomes in the organ transplantations will be based on the HLA disparity between donor recipient pair and presence of anti-HLA antibodies in the recipient. These antibodies could be preformed or de novo in origin, and they have got unfavorable outcomes in the transplants, causing graft failure, delayed engraftment, graft versus host disease, and hence a reduced overall survival rate. This has been well studied and documented in the literature. Click please. So with this, hello, yeah. So with this background, we thought to have an insight in our transplanted patients' outcome with respect to the DSS status. Next please. So we analyzed our patients' data in a retrospective case control manner, and we typed the donors and recipient for the HLA on a high resolution. The checking for the anti-HLA antibodies or the DSA crossmatch was performed on a Luminex 100 flow analyzer and was checked on a Immucor HLA software. The cutoff values of the median fluorescence intensity was taken as positive for more than 1200 and negative for less than 800 and in between as intermediate. We use standard definitions for the critical clinical terminologies, and suitable statistical tests were used for analysis purpose. Next slide, please. So we could see in our study that there are different variables which are responsible for the engraftment or graft failure outcomes in both the categories. However, we could found that DSS status was important and statistically significant in our pilot. Please highlight that point. Click this. Thank you. Next slide. So in fact, out of 42 of our patients, 5 were positive for their DSA status, and this can be traced back to their sensitizing event in the past because they were constantly getting transfusions in the form of red cells and platelets. They had adverse outcome in their transplantations like grade 1 to 3 graft versus host disease and reduced disease-free survival rate. Next slide. So in this first Kaplan-Meier curve, we can see that the probabilities of the fractional survival after 300 days of the post-transplantation in both the groups are falling with the same interval for longer duration, suggestive of a constant survival. We also analyze both the groups for their hazard proportion with this log-log plot analysis, and it is evident from this graph that the first 100 days of the transplantations are crucial because the risk factors are nearly equal in both the groups, and it is increased in case of half-mass transplant compared to the full-mass transplant. We also compared both the groups with respect to their endpoints, versus alive or death status of the recipient. In case of half-mass transplanted patients, which is evident from this bar diagram, that the number of these survived patients in half-mass transplanted are comparably equal to the full-mass transplanted patient. That means selection of an suitable half-mass donor when a full-mass donor was not available based on a negative DSA cross-match status proved beneficial for us to go ahead with the for the good outcomes in the transplanted patients, provided that the survival chances in both the groups are similar. Next slide, please. Our study results are consistently and comparable with the studies in the previous literature, which states that DSA positivity has got deleterious effects in the transplant outcome. The gray area over here to discuss is the MFI cutoff values, which are variable from laboratory to laboratory and from places to places. After all, MFI is a mathematical calculation of an in vitro setup in the laboratory, which is then utilized in vivo for to understand the patient's pathophysiology. Next slide, please. So in a conclusion, Presence of a DSA is not always a contraindication but rather a risk factor. Identification of DSA can make help inform decisions to the clinicians and the choice of immunosuppressions if needed. Donors should be selected as far as possible based on a negative cross-match status of the DSA. In case of a DSA positivity is encountered, one can make an attempt to decrease the antibody titer load using desensitization protocols like plasma exchanges or immunosuppressants or even steroid. What we would like to emphasize over here is it's a need of our in the transplant field for a consensus agreement or standardization of the MFI cutoff values of this DSA, at least for Indian population. No study is a complete study. We do understand certain limitations of a study in like smaller sample size, yet it was statistically significant. Key repertoire was not analyzed here because of the financial constraint, and cytomegalovirus and parvovirus status was not available for all of our patients, which would like to take care in the future extended phase of our study. Next slide, please. Finally, I would like to acknowledge all our patients and their families for the constant belief in us, our peer clinicians and the researchers. Special thanks to the Biotechnica team for providing us a wonderful opportunity to present our study, and special thanks to my mentors for their constant belief and support in me. Thank you, everyone.
Thank you, Anand. Wonderful presentation. Although there were some audio issues, never mind. Over to you, judges. Now you can ask questions.
Yeah, I have only a comment because it's becoming too much. That's a good work, but you spoil it by copying something from somewhere. If you can't make the image, even you write it or draw it in a piece of paper. Don't copy images. At this time, you have to be original 100%. You should not copy a sentence. I hope you never did, but you should never copy an image. You can draw it, or someone can help you draw it or write it. Never copy, because how are you going to create great things when you have to copy something, even if you can't even do that? But otherwise, good work. I don't have questions.
Yeah.
Thanks for this comment, sir. I will take care in the future. Thank you, sir.
Yeah, so that's something, I mean, that's for everybody. I think nobody should actually at this stage do that. A quick question, your survival plot, where was that study done and what was the sample size? And how did it compare to the untreated?
So this... Sir, actually we had transplanted patients for various hematological disorder. There are two scenarios. One is in full mass transplantation, and second is when the full mass donor is not available, which is a very rare situation. So we go ahead with an half mass transplant, that is 50% mass donors. So when we compare outcomes, there are as we know, it is in half mass donor. That means there are certain risk factors. It's not untreated, so they all are treated. Only the thing is the donor is either half match or a full match donor. So we compared the outcomes, whether the DSA holds good or statistically important for the half match.
I guess, Anand, your voice is breaking, and also we have run out of time. So judges, I think we should stop.
I cannot hear you, sir. I beg your pardon.
Yeah, Anand, your internet is not working, no problem. So we'll move on to the next presentation, and that will be in the bioengineering category. Now, before I go into bioengineering category, I have a small announcement to make to all of you. Now, I've been actually reading the comments while the presenters are presenting, and I observed that some of you were sad that I missed the opportunity. So don't be sad. We'll keep coming with more such innovative contests. But we cannot do that unless you go and share this webinar link, the contest link on your WhatsApp, on your Instagram, on your LinkedIn, because if we have more number of viewers who are watching this, then only will be encouraged, our judges will be encouraged to conduct more such contests in the future. Now, quickly moving into the bioengineering category, we have our first participant, Aparna. Hello, Aparna, how are you?
Hello, sir, how are you?
I'm doing very well. So Aparna, over to you. Now you can start your presentation.
Thank you. Good evening, everyone. My name is Aparna S. Narayan, and over the course of the next 4 or so minutes, I will be talking about how this new fascinating technology called brain-computer interface can be used to identify Carnatic ragas. Now, a little disclaimer before I go to the next slide is that unlike the amazing work we've seen so far, this is a review work, so I don't have any observable results or graphs, but I hope to engage in active discussion with the panel members and the audience and build on it in the future. Next slide, please. So Indian classical music is over 5,000 years old, with Carnatic music as we know it today having developed in the 15th or 16th century. Now, there are two main reasons why I chose this particular topic. Firstly, the application of engineering principles into identifying Carnatic ragas is relatively new and literature is sparse. And secondly, I was fascinated by the interdisciplinary approach of this topic, and especially its link to neuroscience. The word raga in itself means color or dye, and it gets this name because it has the ability to invoke emotions in its listener. So a purely physical approach using a computer to analyze what raga it might be will not be enough because it has a biological and neurological component. To carry out this process, I did a preliminary study and understood some concepts of Carnatic music. I carried out a literature survey, critically analyzed said literature, and proposed a broad framework for a multidisciplinary approach in order to build software and products to accurately identify Carnatic ragas in the future. Next slide, please. So I don't know how clear this is to all the viewers, but let me go into it in a bit of detail. So obviously, one way to categorize ragas is to really analyze how people hear it. And we can do this by studying their brain in response to what they hear. And this can be done using EEG or fMRI. It can also be done by monitoring heart rate, which again can be used to study the effect on the automatic nervous system. Now, of course, before engaging in this, we have to make sure that the subjects have sound hearing, don't have any prior neurological conditions, and gauge their level of musical training. Obviously, a musician will be able to identify something much faster than someone like me. Next, the actual audio of the raga can be studied using physical parameters and software such as MATLAB. Now, I'm going to go into two aspects in a little bit of detail. One is that in order to really get to the crux of this issue, we have to separate the gamakas and the non-gamaka sections. Again, this I will come into why we have to do this in the next slide. Secondly, I would like to point out a very interesting new software which calculates something called the raga index calculation. And it predicts the probability of a particular raga based on neural networks. And it's very fascinating foundational work, which will obviously be built upon in the future. Now, we can, of course, having a multidisciplinary approach has many advantages, primary of which is we get results from many sources, and the results hence have more validity. And second, we can build a robust database. Next slide, please. Next slide, please. Thank you. So, as I mentioned gamakas before. Now, gamakas are intrinsic to Indian music. It's when you shake the note. And the problem is when software hear the shaking of the note, they often misidentify the raga. Similarly, ragas which may ascend in a uniform manner, but descend in a non-uniform manner. For example, Sari Gama Padanisa, but descend as Sani Padani Pa Mari Garisa. They also get misidentified often. Further, recent research which has looked at emotional response to ragas have focused on extreme ends of emotions. So they focused on happiness or sadness, but emotions themselves are a spectrum. Finally, different musicians and different instruments have shown to have different responses in all the software tested. All of this is further proof we need a strong biological component. Now, this has a lot of applications and scope, primary of which is to develop software and apps. And now, this is interesting because when you hear a particular raga and you like it, a smart app will be able to recommend songs based on the same melodic structure. Similarly, we can also get new understanding into how our brains listen to songs and how it processes it. And since Indian music in general is very organized, it can be used to classify other genres as well from different countries. Finally, when I was talking to my mentors, they mentioned that this broad framework of using a audio signaling mechanism in brain can also be used to identify other audio stimuli. It may be a language, it may be sounds, but you can understand how our brain processes it and separate it from noise. Finally, there's new research into the impact of raga therapy, which is used for mental health and reducing stress. And this method can be used to really test the efficacy of such methods. Next slide, please. So thank you for your time. Thank you for listening. And thank you for this opportunity.
Okay, wonderful presentation. Very confident, as the audience is also, clapping for your confident presentation. Over to you, judges. Now you can ask questions. Yeah, Dr. Shashidhar, please.
Yeah, very nice topic, very nice tools, and I appreciate your presentation skills as well. Can you measure? Can you quantify the happiness that a person gets by happy chemical? There's some oxytocin.
Oh, yes, definitely. I think so far this hasn't been done. There has been EEG, and there is definite link to happy and sad emotions. But I feel, I didn't go into it, but it was in my slide, that we can also test neurotransmitters, like you mentioned. And there will obviously be changes in levels of dopamine or serotonin in response to different music types. Yes, sir.
Did you do any work at all? Because the topic is great, presentation is good, but there seems to be nothing in it. Did you do any work at all?
No, I mean, this was a review work, sir. I think
we made that disclaimer in the beginning.
Yes, hence the disclaimer. But I think one thing I have added is that all the papers I read, there are physicists working on this, there are electrical engineers, there are neuroscientists, but there is no actual combination of all these things. And I feel like this area will really progress only if you start to combine and take a new direction. So a lot of ideas I have put forth, but it obviously needs extensive practical validation. But yes, I actually did not do any actual lab work.
Thank you.
All right. Thank you. Thank you so much, Aparna and the judges. Okay. So without wasting much of your time, we will move on to the next participant. And Aparna, you can leave the backstage now. Thank you. So our next participant in the same category is Sai Supriya Lakshmi, and she has joined the backstage, bringing you in. Over to you, Sai Supriya Lakshmi.
Am I audible, sir?
Yes, you are.
Yes. Very good evening to one and all present here. I feel very privileged presenting in front of honorable juries, professionals, and staffs who have joined in this live session. So the topic of my work is docetaxel incorporated zinc oxide polycaprolactone nanoscaffold biosensor for lung cancer diagnosis and treatment. And I was guided by Dr. D. Sangeeta. Next slide, please, sir. Yes. So going into my actual work, we all know that cancer is one of the deadliest diseases in the world, and there are many traditional and non-traditional treatments that are done for the treatment of cancer. But nowadays, nanotechnological treatment is getting more importance for the treatment of cancer. And in such a case, we have developed a nanofibrous scaffold wherein we have made use of a polycaprolactone polymer into which the zinc oxide nanoparticles and the drug docetaxel is incorporated into the polymer and being spun as nanofibers. So why we chose zinc oxide is it is already a very good antibacterial material, and in our work it shows cytotoxic effects too. And coming to the drug docetaxel, it is usually a chemotherapeutic drug, and it causes many side effects when used as a chemotherapeutic drug. So we need to reduce the side effects and killing of normal cells in this arena. So we thought of doping both the zinc oxide and the docetaxel into the polycaprolactam solution and obtain the nanofibers. So first what we did is we cross-linked the zinc oxide and the docetaxel into the polymeric solution, and then we optimize the parameters using innovative procedure named central composite design. And then we spun the nanofibers, and these nanofibers were characterized for MTT assay, flow cytometry, etc. The first job of our work is we had to synthesize the zinc oxide nanoparticles. We employed a simple sol gel method wherein zinc nitrate was used, and then ammoniacal and polyethylene glycolic solution was used in order to bring the pH of the solution to neutral. And then suddenly the temperature was raised, and then we obtained the zinc oxide nanoparticles. This was repeatedly washed in order to obtain the zinc oxide nanoparticles. Then we characterize the zinc oxide nanoparticles using SIM analysis. The SIM analysis showed a diameter distribution of around 60 to 70 nanometers. And then we have shown the IRAC spectrum of the zinc oxide nanoparticles. The weight percentage of the zinc and oxygen is as tabulated. And then we went for the spinning of the nanofibrous solution via a technique called as the electrospinning technique. This is the electrospinning setup. The final material we obtained is somewhat like this white color patch-like thing. Next slide, please, sir. Yes. So next, this is the model that we have generated using central composite design. And these are the surface and the contour plots. And from the R-squared value we have obtained, which is above 90%, this showed that the experimental model highly fitted the computational model. And then we went for the SIM analysis for the three types of the nanofibers. For polycaprolactam nanofibers, the diameter was around 166 nanometer, and after the incorporation of the zinc oxide nanoparticles into the polycaprolactam nanofibers, the diameter was slightly increased. That is around 234, and after the incorporation of both docetaxel and the zinc oxide nanoparticles, the diameter is slightly reduced. This is because of the partial congealing effects and the conductivity changes. Then we characterize the zinc oxide nanoparticles for FTIR. Characteristic peak of 443 is obtained, which confirms the presence of ZNO. And then we went for the FTIR spectra of the three electrospun nanoscaffolds also. It showed characteristic peaks like CH, CO, and CC bonds, which are all same for all the three nanofibrous scaffolds, which confirmed its effective incorporation. Also, in order to confirm whether really docetaxel is present into our synthesized nanofibrous scaffolds, we went for a HPLC chromatogram, which showed a peak at a detection wavelength of 214.6 nanometers with a retention time of 11.7 minutes, which pertains to the presence of docetaxel. And then we went for the XRD analysis. This is the XRD analysis of zinc oxide nanoparticles. And this is the XRD analysis of the nanofibrous scaffolds. For polycaprolactone, it was highly crystalline. And after the incorporation of zinc oxide, it turned amorphous. And after the addition of both zinc oxide and docetaxel, a semi-crystalline nature was seen. And this is highly needed for anti-cancerous applications. We also characterized it for TGA and TSE.
Your time is up. Sai Supriya Lakshmi, your time is up.
Sir, next slide, please, sir. Next slide, sir. Final slide, sir. Yeah, this is the actual studies we did. MTT assay showed an IC50 value of 61.8, which is very much good. And then it showed a hemolysis percentage of 4.47, which is very much less than 5%, enabling it a good biomaterial. And it became a very good apoptotic marker.
All right, your time's up. So over to you, judges, if you have any quick questions.
Yeah, I was just eager to hear your later biological assays, but the time was short. But maybe next time you can focus more on that. It's okay.
All right.
Yes, sir.
Okay. Any more questions? Okay. Looks like there are no more questions. Thank you, Sai Supriya Lakshmi, for your time. It was nice listening.
Yes, sir. Thank you, Biotechnica. Thank you so much for this opportunity.
Okay. You may leave the backstage now. So judges and my dear audience, we are running out of time, so we have to be really quick. Okay. And now we have our next presenter, and she has joined us now. Over to you, Siva Darshini.
Very good evening to another. Okay, sir. Thank you, sir. Very good evening to another person here. I am here to present my topic, utilization of alginate from brown macroalgae for ethanol production in Clostridium phytofermentans. Brown algae is a very good feedstock as compared with other feedstocks like rice or any cash crops, because it has a high amount of carbohydrates contains in it, and it have a very profitable way. You don't want any arable land or water supply to cultivate it. And brown algae is need the effective microbial investigation to make it useful for the bioethanol production, because we have the in the composition. So next. In the composition of brown algae, we have lots and lots of carbohydrates. We need the many effective microbial strains to convert them into a profitable bioethanol. In this study, we studied Clostridium phytofermentans. Clostridium phytofermentans is a versatile microbe. In this microbe, we can see the 3 different alginate lyase gene-specific enzymes, and these presence of these metabolism, we can use the Clostridium phytofermentans as the efficient microbe to utilize it for bioethanol production using brown macroalgae as a substrate. And we examine the brown macroalgae. We examine the Clostridium phytofermentan can grow in the alginate medium without any inhibition. So we use the microscopic analysis, which all the samples are stained with cyto9. And in the picture A, we showed the stained samples without Clostridium phytofermentans, and B and C are the stained samples with the presence of Clostridium phytofermentans grown on the synthetic sodium alginate medium, and it showed the different. It shown the different time period, like 24 hours of incubation and 48 hours of incubation. And in the some images, we showed the in the picture of D, we showed just the some image of synthetic sodium alginate media without any inoculation of Clostridium phytofermentans, and E is showing the absolute assimilation of synthetic sodium alginate by the presence of Clostridium phytofermentans incubated for the period of 120 years. And it is visualizing the potency of the Clostridium phytofermentans. It can easily assimilate the alginate present in the brown macroalgae. So next slide, sir. In the case of brown macroalgae, we took the commonly used brown macroalgae identified from the hostel areas, and we subjected for carbohydrate composition as compared with other carbohydrates like cellulose, xylene. Alginate content is very high. Alginate content is very high, so we need to assimilate the alginate content. So only we can use it beneficiary for the production of ethanol, because as comparatively cellulose, xylene, Mg, xylene, everything, alginate content is too high. So, and we did the FKF spectrum for alginate extracted from these two common brown macroalgae, and these spectrum peaks are showing the characteristics of same synthetic sodium alginate. And this visualizes that sodium alginate extracts from the common brown algae have the efficient amount of alginate present in it. And we subjected the Clostridium phytofermentans to know about the growth pattern of Clostridium phytofermentans subjected with the different supplements of alginate in the fermentation media of GS2. It is commonly used for Clostridium fermentation, and we did the alginate. We used the synthetic sodium alginate as the control group, and we subjected the Clostridium phytofermentans in the media contained supplemented with alginate extracted from Pantina tetrastomatica and alginate from Terminaria ornata, and these growth patterns showing different with different supplements of alginate substrate. And we conducted the batch fermentation of ethanol for the ethanol concentration and ethanol productivity of brown algae using the Clostridium phytofermentans. It's visualizing that sodium alginate is a control group, and we used other alginate extracts from Pantina tetrastomatica and Terminaria as the main samples, and we conducted the fermentation method like
Shiva, your time's up. Okay.
Sir, 1 minute, sir. And we did the fermentation for all 3, and we can see the ethanol concentration. It is increased after 172 hours.
Over to the judges. They can answer the questions. Yeah. Over to you, judges. Do you have any questions?
Yeah, just a comment. Just like the previous presentation, you should never, minimize your time for the final study, final impact, right? Ethanol presentation
Yes.
has been the goal, so we couldn't hear that because of your
Sir, I
balanced
answered.
presentation. But it's okay. Yeah, thank you.
Okay. So thank you, Shivadarshini. Your presentation was really good, but yes, time ran out, no problem. So you can leave the backstage now. And now we reach the 8th category out of the 10, and that is medical devices. Now, medical devices, as always, yes, medicine, medical devices, very important category. And to present the first one in that category, we have Shubham Gupta. Shubham, how are you? Can you hear me? You need to unmute your mic. Okay. Yeah. You can start the presentation now.
Am I audible?
Yes, you are audible.
Yeah, thank you so much, sir, for a very good evening and a warm welcome to this evening of Biotechnica, in which we are going to share some innovative ideas on various fields. So, I, Shubham Gupta from King George Medical University, going to present my poster on the medical device category of photography for screening various embryogenic conditions in children. So, proceeding ahead, I just want to tell you that we are just already know, due to this pandemic, we came to know that day by day our eye vision acuity is getting reduced, and we are facing a lot of pressure in our eyes. Our traditional way of working has now been shifted to the digital plethora. That is the reason, and therefore my study attempt to validate this smartphone photography for screening various kinds of embryogenic conditions, especially in children, but we can also go for the adults also. So proceeding ahead towards this study, as I just want to know, before early 3 to 5 years of children has been photographed by an optometrist for capturing their pupillary red reflexes, and this is also for the next, followed by clinical examination by the principal investigator, who is a physician, and that physician, on the basis of those clinical examination like retinoscopy, identified the children with various significant embryogenic conditions, right? Subsequently, on other hand, two other ophthalmologists independently, ophthalmologist 1 and ophthalmologist 2, as you can see in this result chart, they are categorizing those photographs on the basis of the color, reflex, symmetry, shape, and the pupillary reflex into two categories, like abnormal and the normal category. Ophthalmologist 1 collecting data 2, and ophthalmologist 2 is collecting data 3rd. So, and clinical examination is a data 1. Please note this. So moving ahead, look in this methods, you can see data 1, which is the clinical examination, will then compare to that on the photography with the data 2 and data 3. I just want to include this thing, comparing these data 1 with data 2 and 3, because for your verification, for your accuracy, if someone is having any doubt that the clinical examination is not okay, I'm not satisfied, then he or she can go for them for photographs also and verify that this is my ocular problems. As you can see in this all 10 photographs, there are certain different kinds of embryogenic conditions which are going on, and patients, there are a lot in numbers. So finally, moving step towards a result, there are total 250 children has been screened clinically, out of which 59, means 23.6%, were having and harboring embryogenic conditions. So, which mean sensitivity and specificity of a screening by smartphone photography were 94% and 91% respectively, right? And with this, want to include one more thing, that the agreement between the PI and the ophthalmologist one was 83.2%, and ophthalmologist 2 and the PI agreement is 87.2%. So this is an accuracy and sensitivity which we are getting through the smartphone photography, and hence we can say in the conclusion that smartphone photography is going to be play a pivotal role in the field of telemedicine, whether you talk about the e-Sanjeevani, which is the telemedicine hub of the government. This is also going to play a significant role in this in screening embryogenic conditions in children especially. And before concluding my speech, I just want to share one quote. I don't want to recall the name.
Okay.
You might.
I believe
want to be
Shubham got
remembered
disconnected.
as someone
Shubham, can you hear us?
who put India on the scientific map of the world in terms of
Okay,
the large
guess he
innovation.
got disconnected.
Yes,
We'll just
yes.
wait for few seconds. Okay.
Shall I proceed?
Okay. All right.
So through
Strangely,
this
I'm
presentation,
not
I
getting
just
any audio
want
from him.
to share. So with this, hello, yeah, am I audible? Yeah.
Okay, looks like there was a connection issue. Yeah. Over to the judges, if you have any questions.
Right. Subham, this work that you presented by one slide, good, you just kept it to one slide. Is this your work or is somebody else's work? I don't know whether Subham can hear me, Sekhar, or maybe Sekhar is again gone.
Can you guys hear me? Hello?
Yes.
Yeah, we can hear you.
So looks like Shubham is having internet issues. Okay.
It was obviously somebody else's work. He just presented the paper from somebody else. I gave the link.
Yeah, that's what I was also, because I couldn't read the whole of the poster. Want to know, it was an interesting work, but whether how much original work was it, those things I couldn't really go through it. Okay.
You can
But
see
now
the link. I sent.
Yeah. Okay. Okay.
Okay. All right. So quickly moving on to our next presenter, and that is Athira. So Athira, can you hear me?
Yes, sir. I can hear you.
Okay. So Athira belongs to the medical devices category. Second presenter, over to you, Athira.
Good evening, all. I'm Athira Srinivas from Manipal Academy of Higher Education, and my topic is minimally invasive tissue sampling, myths, and alternative to conventional autopsy in neonatal deaths. Can I have the next slide, please? According to UNICEF, the neonatal mortality rate globally was 18 deaths per 1,000 live births, and India had an average account of 23 deaths per 1,000 live births in the year 2018. This is 9 times larger than from the developed nations, which was 3 deaths per 1,000 live births. India shares a quarter of the neonatal mortality rate India shares quarter of the neonatal mortality rate, and for accurate diagnosis of the fetal, perinatal, and neonatal autopsy, we require a gold standard method, which is known as conventional autopsy or post-mortem examination. This will help in confirming and establishing the cause of death in the neonatal period. Even though this technique is a gold standard, there are challenges in conventional autopsy. India being a conservative multicultural nation, the refusal to consent by parents due to public reluctance, adverse media attention, delay in last rites, religious and cultural beliefs, fear of disfigurement of deceased body, or emotional attachment are the main reason for them to refuse the conventional autopsy. Can I have the next slide? To overcome this situation, a new technique called minimally invasive tissue sampling, or MITS, was developed by Barcelona Institute of Global Health in the year 2013. This technique collects the tissue samples from relevant organs and body fluids using fine needles soon after death. It is a very feasible, less traumatic, and less time-consuming technique which can circumvent the need for a complete autopsy in establishing cause of death. The tissue samples collected can be taken for histological, microbiological, as well as for molecular examination. The picture shows barbed monopty needle which is used to perform the test. Can I have the next slide? This is the needle in which it have very sharp tips which will be able to collect core samples, core tissue samples with a polished surface which ensures smooth entry and exit into the target organs. It is also lightweight and very ergonomic to handle. It consists of angled bevels which ensure that there is minimal crush artifact for the samples that we are collecting. Also, it consists of an angled deep notch sample which ensures the high core that we collect are high qualified. And the green end area, we can see it is rotatable in a clockwise manner so that it ensures that the collected samples can be easily removed from the needle to the sample container. The second picture shows that collection of liver biopsy, liver tissue from a neonate using this needle. Can I have the next slide? Literature shows 100% concordance between conventional autopsy and minimally invasive tissue sampling, so that we can consider this as a replacement for conventional autopsy. The main outcome of the study is it can establish cause of death and it can change the perception of the community on fetal autopsy. And there are benefits for the society, like in a socially constrained area where they refuse fetal autopsy, we can take this method, minimally invasive tissue sampling, for a convenient replacement. And also, this is able to establish cause of death. This can prevent deaths and morbidity in subsequent pregnancies. Also, this can improve the neonatal mortality rate, and this can aid in recommending health policies. And there is no delay in last rites, and no religious and cultural beliefs are being affected. Since this can be considered as a feasible replacement, there is no disfigurement for the body, where the family can take the deceased body back for their cremation and religious purposes. And finally, summarizing this talk, I would like to advise minimally invasive tissue sampling as a convenient replacement for conventional autopsy, where it is a very socially constrained. Thank you.
All right, so time's up. Over to you, judges, if you have any questions.
Since it's autopsy, why are we worried about minimally invasive? We are not looking at live people, no? Autopsy is always invasive.
Yes, sir, it is not. But while we are approaching parents for to correlate with because literature has shown that there are scientific, the certified cause of death and actual cause of death varies. And mainly clinical specifications are not same. So while we approach parents for leaving the baby's body, they are refusing. So if we are considering this technique, we can take the samples as well as we can give the body back to the family, so that even in subsequent pregnancies, may not be a genetic reason. For genetic reason, we have many techniques, but if it's not a genetic reason and there are many other problems what the babies are facing, so that we can take the samples and can correlate with the clinicals, and later we can get back to the parents for a clinical advice for a subsequent pregnancy, for the management of the future pregnancy, sir.
So, Athena, sorry, Raja,
Yes,
you
sir.
want to go ahead?
No,
A quick question. The device that you showed,
yes, sir.
Did you make it? Did you prototype it? Where was this taken from?
No, sir. This was actually developed by Barcelona Institute in the year 2013. We are actually working on new needles in which the term and preterms, because there actually the consistency of the brain tissue is different for those, so that we are actually looking for more facilitated instrument for that. In India, this work has not been yet done. This needle has not been used till now.
Okay, this might be a little, you've shown the device, so I'm therefore impelled to ask you this query. What is the regulation of that device itself? And it will be very interesting to know because it's for post-mortem analysis. So how do regulatory agencies look at such devices?
Sir, I didn't get that question, sir. Regulatory agents means
Okay, no, that's fine. I just wanted to gauge that side of the device, whether you have studied the regulatory process of such device, especially those going to be involved in post-mortem analysis, and what kind of ethical consideration are taken into account for such devices.
so before this process, we will be taking a consent, and also the participant information sheet will be signed from the parents of the baby.
Okay.
We make sure that.
Shekhar, we have 30 seconds. If somebody else wants to go, I have one small, again, query. So the needle that you are trying to prepare in your lab, how do you differentiate that? Are you using a different kind of titanium needle, or what are the constructs of those needles, and how it is different?
Sir, like only on the bevel part and on the area of the sample notch, we have to make it different. I mean, we have to make it more sophisticated so that the brain tissues doesn't get damaged. Nowadays, we are getting very fragile tissues. So we are in a plan to make it, sir. The body, everything will be titanium only, but only the bevel part, the notch side where the cores will be accompanied, there only, sir.
So are you going to file a design file?
Yes, sir. If we can do that.
All right.
Thank you.
Thank you.
Thank you, Athira. We're running out of time, so quickly. Now we'll move on to the next participant, Priyanka Shah from the medical devices category. Priyanka, please come in. Yeah, over to you, Priyanka.
Okay. Good evening, everyone. I am Priyanka Shah, and on behalf of my team consisting of Ranjani, Saivi, Sabista, and Dr. Kushagra, I will be discussing about a novel technique which is a 3D colorimetric method for COVID-19 detection. COVID-19 is the worst pandemic that the world has witnessed in the last century. Next slide, please. So how is the world combating with COVID-19? Nasal and throat swabs are the most common technique used for detection of COVID-19. However, it is a painful process. Even dogs have been trained to detect COVID-19 patient. Now let's talk about our proposed device, which is a breath analyzer. Breath analyzers are usually non-invasive, and it is also suitable for identification of asymptomatic and pre-symptomatic individuals. Any diseased person generates volatile organic compounds or VOCs from the exhaled breath. So what are VOCs? They are simply chemicals that have high vapor pressure at room temperature. Our proposed device consists of elastomer layers which can either be hydrogel or PDMS. Each of these layers are embedded with a particular type of porphyrin. Porphyrins are a group of heterocyclic macrocyclic organic compounds. You would be amazed to know that the basic structure of chlorophyll molecule is actually a porphyrin ring, which is responsible for the green color in plants. By measuring the VOC profile in elastomer matrices, we are able to identify unique VOC fingerprints associated with COVID-19. You must be wondering, why should we use this device, or why is it better than the already existing one? Next slide, please. Advantages of this device are manifolds. It has easy visual analysis, it is inexpensive, it not only gives quicker results, but it is also non-invasive, user-friendly, and can be used for mass testing. Next slide, please. The first step involves a person exhaling into the breath analyzer. It has been observed that ethyl butanoate is found in high concentration in COVID-19 patients. When an infected individual exhales into the breath analyzer, ethyl butanoate reacts with the porphyrin present in the elastomer layer. In our device, we have chosen 8 porphyrins that react with ethyl butanoate. The porphyrins are initially green in color, and upon reaction with ethyl butanoate, it turns into red. This change in color of the sensor will indicate with an accuracy of 90 to 95% that the concerned individual had COVID-19. It is to be noted that the presence of ethyl butanoate is also observed in people having upper respiratory tract infection. However, the amount observed in COVID-19 patients is comparatively higher than that in URTI patients. The mean value of the quantity of ethyl butanoate found in URTI patients can be taken as a baseline for COVID-19 detection. Next slide, please. Thus, to conclude, our proposed device has the potential to curb the spread of coronavirus by introducing a rapid, non-invasive mass detection method. Thank you. I would like to thank Biotechnica for giving us the opportunity to present our novel idea.
Okay, thank you, Priyanka. Judges, if you have any quick question for Priyanka.
So how far you have gone to produce it or make a prototype? Where are you in the?
Sir, we have studied the literature, and from the literature, we have derived that what are the VOCs which are associated with COVID-19, and what are the porphyrins that may react to COVID-19.
Can you tell us one paper that you remember you read and that you were impressed about the work when you were doing?
Yes, sir. Yes, sir. I was impressed by Suslick's paper, and he actually the use of porphyrins to detect VOCs was an idea of him.
Okay, thank you.
Okay. So with this, we come to an end of this category. Thank you, Priyanka. You may leave the backstage now. Moving ahead, we have our next and the 9th category, microbiome. And to start the microbiome category, we have with us Kusuma. So Kusuma, over to you if you can hear me. Can you hear me?
Good evening, everyone.
You can start.
Am I audible?
Yes, you are audible. Please start.
Good evening, everyone. Myself, Kusma. I'm working at National JAMA Institute for Leprosy. I'm working on Mycobacterium tuberculosis. After the inhalation of Mycobacterium tuberculosis through the droplets from the infected one, it will directly enter into the lungs. At the lungs, the macrophages identifies the bacteria and it engulfs the bacteria and it will try to eliminate it. Our immune response is very efficient that it will be eliminate the whole bacteria even if we don't take the chemotherapy also. But it will take a long time. It can't even eliminate the whole bacteria completely. So within this time, the Mycobacterium tuberculosis will escape from the macrophage and it will disseminate into the bloodstream and other peripheral sites like liver, spleen, and the lymph node. There are so many factors that are responsible for the dissemination of the MTB. Depending upon the strain of the MTB and gene polymorphisms. Here, I'm mainly focusing on the vascular endothelial growth factor, and the ELR positive chemokines, which are also known as the angiogenic chemokines. The human necrosis factor alpha is responsible for the secretion of these both the factors, VEGFA and ELR positive chemokines. As the part of the immune system, immune response, the whole T cells will migrate to the infection site and it will form a granuloma with very high vascularity. Here, our main study is, our hypothesis is that if we regulate the vascularity, can we increase the drug penetration at the granuloma site, and we will be able to decrease the bacterial count. And even if we decrease the vasculature, maybe the dissemination of the MTB might be regulated so that we can regulate the dissemination of the bacteria from the lungs to the other sites like liver, spleen, and the lymph node. Next slide. To approach this objective, we classified so many groups of the guinea pigs. Firstly, we inhibited the soluble TNF because we didn't choose the whole TNF because the TNF is the main factor that is involved in the immunity. So part of the soluble TNF we are inhibiting here. We used XENP 1.95 that will go and bind to the TNF factors and eliminate binding and signaling through the TNF receptors. And these are our results. We have seen these are the fold changes that are compared with the control group, and you can see the high expression of the ELR positive chemokines from the compared to the 4th week. There is high increase at the 8th week. By inhibiting the TNF, we decreased the ELR positive chemokines without any compromise in the host immunity. At the 4th week, we saw the changes in the migration of CD4 plus and CD8 cells, but at the 8th week, we didn't see any compromise in the immunity by using this inhibition of the soluble TNF. Next slide, please. Here, even after the 8th week, we didn't even found any bacterial count at the liver and the lymph node in the treated group. When compared to the control group, we found the bacterial dissemination even at the liver, lymph nodes, and you can see here we mentioned the CFU log values. Here there is a high bacterial count at the control group when compared to this treated group. As a part of our study, after that we will be inhibiting the vascular endothelial growth factor and one of the ELR positive chemokine receptor, which is the main receptor that we have seen in the study, which expression is very high. We will be inhibiting these before the MTB infection and as well as the after MTB, after the granuloma formation. Along with the chemotherapy, we will be giving these drugs because any patient will not be able to take the drugs before the infection itself. So to find out the role and as well as its functions, we will be.
Okay, your time's up. Kusuma, your time's up. Bringing in the judges. Yeah. Over to you, judges. The question session. Do you have any more questions?
So what was the rationale in choosing guinea pigs here?
Because in the mice we can't see the granuloma formation. Guinea pig is the main model that we can see the granuloma formation clearly.
People commonly use that model, is it? I'm not aware of it. Most of the studies are done in guinea pigs only for tuberculosis.
Because the granuloma is not formed in the mice, only nodules are formed.
Okay.
Yeah. Good. Thank you.
All right. Thank you, Kusuma. That was wonderful. Moving on to our next poster presenter, and that will be Deepansh Modi, M4, poster ID number. Deepansh, can you please join in? Okay, we have Deepansh here. Deepansh, your time starts now.
Good evening, everyone. I'm Deepansh Modi, and I'm here to talk about my proposal to deal with. If you could change the slide, please. Yes. So I'm here to talk about my proposal to deal with consequential comorbidities of COVID-19 amongst the patients of cardiovascular diseases. With a near 12% fatality rate, these comorbidities pose a major concern. And the present line of treatment, the ACE inhibitors, they reduce cardiac hypertension by reducing vasoconstriction by preventing the formation of angiotensin II, but they have also been reported to increase ACE2 receptors on lungs and blood vessels, which are the binding sites for the novel SARS-CoV-2 virus, and it facilitates their entry into the host cell. So as an attempt to address this issue, we propose to exploit the interplay between gut microbiome and microRNAs. As they say, our body is an ecosystem which needs to be preserved. Gut is a very crucial component of this ecosystem as it hosts trillions of microbial species, eubiosis of which plays a major role in maintaining the health of the host. These microbial species also metabolize our ingested foods to liberate metabolites, which are involved in modulating the expression levels of hosts and other microbial communities' genetic materials. And while only 3% of our DNA gets expressed, the remaining 97% transcribes as non-coding RNA, and short sequences of 18 to 25 nucleotides of these non-coding RNAs are called microRNAs, which disseminate throughout the body to different organs to regulate expression of various genes encapsulated in endovesicles. So moreover, an atypical miRNA expression level is also a biomarker for several disorders, and their normalization does hold the potential for proposing a novel therapeutic strategy. Can we have the next slide, please? To address the problem at hand, we got a few leads to edify our hypothesis. The genesis of our idea was inspired by a recent ICGEV study that pointed at an alanine to valine mutation in the 930th amino acid of Indian SARS-CoV-2 virus's spike protein. This mutation rendered the virus more prone to attack by a host-produced antiviral microRNA, which is the MIR27B, and it prevented the cell from binding to the virus. The virus from binding to the cell. Moreover, MIR27B had previously been found to downregulate the expression of ACE, which shows its involvement in cardiovascular system. On digging more about the MIR27B and scanning for its targets, we found it to be involved in regulating myocardian expression, which is a transcription factor that is involved in smooth muscle cells differentiation in the cardiovascular system. Next, we searched the MIR base and realized that MIR27B is actually a gut-derived microRNA, which makes it a fecal microRNA, and it had a positive correlation published with the genus Enterococcus. So we next dug for drugs or dietary products which had the potential to enhance enterococcal species in the gut and modulate the levels of MIR27B. Next slide, please. So in conclusion, we propose that our hypothesis is edified by MIR27B being a host and produced antiviral microRNA that attacks the SARS-CoV-2 virus, reducing its infectivity. It also regulates the expression of ACE and myocardian, both of which are involved in cardiac hypertension. And moreover, MIR27B has a fecal dependence, which means it is derived from the gut, and it is also dependent on enterococcal species, which we can modulate by our design diet. Therefore, we propose to exploit this interplay between gut microbiome and microRNAs to target COVID-19 as well as the related cardiac hypertension by modulating MIR27B levels with a diet that we can design. I would like to thank you all for giving me this opportunity and for being patient while I presented.
All right. Over to you, judges, for the questions.
How do you jump from in silico prediction of microRNA interaction from the microRNA produced from a gut microbe to some other tissue, something else is happening? How do you...
microRNAs have this ability to get transported inside the endovesicle. So they can act like hormones and get transported.
So is there evidence that something going through the blood is one thing, from the gut to, for example, respiratory tract? Is there any evidence?
Yes, sir. So the gut has various access to different organs of the body. There is one that connects to the brain.
Tell me one paper where there was one article or review you have read where from the gut microRNA has gone to respiratory system?
Sir, respiratory system, it all, well, not necessarily respiratory system I read, but
or any other system?
Yes. For example, since I am working on cardiovascular diseases, so I know a little about how, miRNA from the gut it was not MIR27B, but another microRNA got transported to the cardiovascular system, and it was involved in atherosclerosis.
There is a report like that. Okay. But do you think that the in silico prediction has to be tested in terms of the actual interaction also first?
Yes, sir. So that's a major drawback of mRNA related therapeutics right now. That the in silico prediction differs from how it actually functions inside the body. So while that is a challenge, but it doesn't stop us from investigating more into it. Perhaps we could get through it.
This is the starting point, all right. But you do realize that has to be investigated. Okay.
Yeah.
And it's a simple experiment to do.
All right. Thank you, Deepansh, for your time. Thank you, judges. Yeah.
Just a second. Very well presented. Well done.
Thank you, sir.
Thank you,
well
sir.
done.
Thank
Well
you.
done. Well done.
Thank you so much.
Okay. So now quickly moving on to our next category, which is marine biotechnology, the last category. And the first presenter in this category will be poster number MB17, Sahana Ghosh. Sahana, are you with us? Yes. So over to you,
Yes, am
Sahana.
I audible?
Now. Yes, you are audible.
Okay.
Go ahead.
Good evening. I am Shana Ghosh, a postgraduate student of Calcutta University from the Department of Zoology. Today I am here to present my poster, which is basically on my review work. It's not my own work, it's a review work. And the topic is the role of marine organisms in the production of biomedicine compounds. Now, ocean is known as the mother of origin of life, as life has started in the sea. Marine biotechnology, which is also known as the blue technology, processes various marine organisms and creates products by the application of various biotechnological techniques. But many of these products have pharmacological activities. Various marine organisms are there which can be used in the production of bioactive compounds. And these bioactive compounds have antibacterial, antifungal, and anti-cancerous properties. So due to this reason, these compounds can be used in the treatment of some deadly diseases like cancer, AIDS, and some types of arthritis. So today I'm going to discuss about few marine organisms which can be used in the production of some bioactive compounds. Next slide, please. So at the beginning, I would like to say that I have kept the slide very simple, and I will give you examples of the marine organisms which can be used in this field in the production of bioactive compounds. So the first organism is the Alteromonas. It is a marine bacterium which has been found to produce a microbial metabolite which has anti-HIV properties. That means this particular microbial metabolite can be used in the treatment of HIV. There are certain marine seaweeds, including red algae, green algae. I've already mentioned the name like alba, then spherococcus, which have been found to produce certain compounds which have antibacterial, anti-inflammatory, and anti-tumor properties. Next is Darcitus. It's a marine sponge which has been found to produce a compound darcetine, which has cytotoxic activities, and it is very much active against B16 melanoma cells and lung carcinoma. Next is Montipora, which is a marine cnidarian, which has been found to produce 3 types of diacetylene. And all these 3 types of diacetylene have been found to produce certain effects which have cytotoxic activities, and those cytotoxic activities are against a range of tumor cell lines. And last but not the least is Chlomochlorus cavi, which is a marine mollusk. It has been found to produce a compound called chromodurolite A, which has cytotoxic activities and at the same time the antimicrobial properties. So next slide, please. So in the conclusion from all these examples, we can say that marine-derived pharmaceuticals can be used as a source of chemical diversity in the drug discovery program. And there lies the application and the advancement of marine biotechnology in the discovery of biomedicines. So in this way, my review work describes the role of marine organisms in the production of biomedicine compounds. Thank you for your patience hearing.
Thank you, Sahana. Over to you, judges, if you have any questions quickly.
You did any work or it's just a literature review?
It's a literature review.
For the review, yeah.
Thank you.
So no question from my side. Thank you.
All right. So thank you, Sahana, for your time. Wonderful presentation again.
Thank you for the opportunity.
Thank you. All right. So we'll move on to the next poster, and that is from Kanika. Kanika will join us shortly. Yes, Kanika is here. So over to you, Kanika.
Am I audible, sir?
Yes, you are. Please.
Good evening, sir. I'm Kanika Arora.
Today, I would like to talk about how to exploit microalgae to live life more sustainably. So, next slide, please. So the question is, why microalgae? As we all know that freshwater reserves are very limited and its integrity and environmental perturbation should never be compromised. Every year, about 3.4 million people died and many suffered from plethora of waterborne diseases due to reckless discharge of wastewater from industries and even from domestic waste in the STPs. This wastewater contains large amount of nutrients like nitrates and phosphates, which lead to algal bloom and consequently eutrophication and degradation of limited freshwater reserves. So what if we introduce microalgae into the picture just before the discharge? That is in the form of a tertiary biotreatment. Microalgae provided with the optimum condition will consume nitrates and phosphates and also reduce the BOD and COD levels. It will not only remove the heavy metals like zinc, cadmium and mercury, but also reduce the obnoxious coliform bacteria present in it. So the end product will be algal biomass and the clean water. This process seems to be quite simple, isn't it? But actually it is not. So next slide, please. So the how. Microalgae can be cultivated in open ponds or closed PBRs, that is the photobioreactors. They give more of a control to grow the algae in a more controlled and regulated manner. Or it can be even advanced setup like integrated systems, which are the hybrid of open and the closed PBRs. There are many factors which affect the microalgae growth, like pH, temperature, light intensity, nutrients, salinity. After the harvesting and the processing, the end products could be biofertilizers, exopolysaccharides, biofuels, and even biopolymers, depending upon the strain used. For example, Botrygococcus brunei is the algae which has a high lipid content. So it can be used for biofuel production. The next slide. So the question is how? What is this holistic approach all about? Every nation needs to achieve sustainable development goals to have a sustainable economy. And microalgae have enormous potential to realize all the 17 sustainable development goals and deeply impact the environment. Like SG Goal 2, which is zero hunger. Microalgae are very rich in proteins and PUFA and many other nutrients. Therefore, epitomize high nutrition diet and also help to eradicate malnutrition from the country. Like SG Goal 6, like the clean water and sanitation, as we have already discussed the phycoremediation of wastewater using algae. Then SG Goal 7, which is affordable and clean energy. Microalgae is a perfect substitute for petroleum. So it can harness the production of biofuel and even bioelectricity. Then SG Goal 13, which is the climate action. As we all know that microalgae consume large amount of carbon dioxide during photosynthesis and in turn produce about 50% of the Earth's oxygen. So in turn, microalgae is a major carbon sink for all the greenhouse gases, and it also aids in curbing air pollution. Despite all the lucrative services, challenges also persist. There are many challenges like large-scale production, which is due to high cost, heavy nutrient load, harvesting algae from treated water, and downstream purification. So in the end, I would like to conclude that microalgae-based wastewater treatment can be an effective biorefinery approach towards a more sustainable and circular bioeconomy. Thank you.
All right. Thank you, Kanika. Over to you, judges. Yeah.
No questions from me.
Any questions? Okay. Looks like there are no more questions. Thank you, Kanika. Thank you for your time. Okay. So now we will move on to our next participant, and that is Vaishnavi. So Vaishnavi is poster number MB8, and now I'll add her in. Yeah, over to you, Vaishnavi.
Sir, can you hear me?
Yes, we can hear you.
Hello, sir. Am I audible?
Yes, you are audible. Please proceed. Yes, you are audible.
Good evening, one and all. My name is Vaishnavi Bomakanti. I'm an undergraduate student from Mumbai University. When we talk about marine bioactive molecules, most of the reviewers limit their study to sponges, algae, cyanobacteria, and microorganisms. So I thought of specifically highlighting the importance of phylum Cnidaria in pharmaceutical industry. Cnidarians are aquatic invertebrates that include sea anemones, hydra, jellyfish, and corals, etc. The common thing among them is the venom that contains neurotoxic peptides which can block the voltage-gated sodium and potassium ion channels, which could be used for treating some of the neurological disorders. In fact, one of the neurotoxic peptide, SHK186, is already out in the market under the name dalazitide for the treatment of multiple sclerosis. Cineraria exhibits neuroprotective and anti-inflammatory property which could be used to speed up the recovery after spinal cord injury, while the rest inhibit the settlements of benthic diatoms and the bacterial attachments. Synthetic corals enhance the regeneration of bone, and recently a coral wedge has been used to treat the valgus deformity of the knee. Next slide. Thank you. The norm from Nemopilima shows anti-tumor effects in the xenograft mouse model and thus could be used in cancer therapy, while the collagen from Rupilema, it kind of shows tissue regeneration in mice models, so it could be used in dealing with wounds. Collagen again can reduce the oxidative stress in tissues, and the norm has got analgesic effects. GFP could be obviously a less toxic and less expensive alternative for radiolabels. Extracts from Anthophylla shows antibacterial as well as antifungal activity and could be used in designing antibiotics, while the rest show anti-hemorrhage properties. Dr. Sunagar from IISc has made the statement that a multi-step genetic process that is involved in the formation of stem cells could inspire a new way of delivering drugs into human system. Interesting, isn't it? But not many people know about this, so PG students who are looking for a new project, this topic is a good one to start with. Next slide. Yeah. You'll be surprised to know that Nematostella vectensis, one of the sea anemone, such a basal animal, has got high number of genes underlying human diseases. In fact, some of the ancestral features of human genome that are now missing in C. elegans and Drosophila are also found here. So if you're looking for a cheaper model over the vertebrate models that we use, this model is a good one to start with. Mylipora could neutralize the blood clots formed by snake venom, and thus it could be used as an antivenom for snake bites. So it is clear that the Cnidarin products have got a bright future in pharmaceutical industry. However, only 3 classes have been focused majorly, so there is a need to explore the organisms, the Cnidarians from the other classes as well to get the best out of this phylum. Looking at these properties, I have mentioned few points that I feel could be the applications of the Cnidarin peptides. For example, the antioxidant property could be used in designing anti-aging creams since we know that the primary reason for aging is the oxidative stress. While the anti-inflammatory property could be used in designing prickly heat powders since these kind of leave a soothing effect on the skin. The antifungal property could be used in designing anti-dandruff shampoo since a fungus known as Malassezia triggers dandruff in the hair. While the antifouling property could be a best solution for treating biofouling problems in aquacultures, since these fouling communities, they kind of interfere with the operation of valves and also compete with the fishes for... Okay.
Yeah. So over to the judges. If you have any questions.
No questions from me.
It's a good one. Thank you.
Thank you, sir. Thanks a lot.
Good presentation, Vaishnavi. Well done.
Thanks a lot, sir. Thank you. Thank you, Biotechnica, for giving this opportunity.
Thank you so much, Vaishnavi. Now moving on to the last presentation and in the Omics category. And this is today's last presentation by Ambreen. So over to you, Ambreen.
Thank you very much. and welcome to my presentation. So basically, we all know how painful it is to have an injection when the doctor gives us. Now think of the situation when people undergoing liver biopsy feel. I'm talking here about liver biopsy because it's the only gold standard available to diagnose the disease called non-alcoholic fatty liver disease or NAFLD. As the name goes, it is a liver disease caused by the accumulation of excess fat in the liver due to conditions not associated with alcohol consumption. This is currently the most common cause for chronic liver diseases worldwide, and it has already affected about a quarter of the world's population. It's currently increasing in children, adolescents, as well as in adult population. So we clearly are in need to identify new non-invasive diagnostic methods, and one option would be to identify diagnostic biomarkers. For this, I use metabolomics coupled with machine learning. Can I have the next slide, please? So metabolomics basically is the study of very small molecules called metabolites inside the cells or tissues or even organisms. So this approach is quite straightforward and begins with the sample preparation. So the samples could be anything from plasma or urine, stool, or any biological tissues for that matter. So these samples are prepared in such a way that they could be read by the instrument, which in our case would be the liquid chromatography mass spectrometer. For comparison purposes, I have taken plasma samples from healthy individuals and those who have been affected by the disease. So the data coming from the LCMS instrument is then further analyzed and then the identification process happens. However, I would like to tell you that this process can be really time consuming. And so in order to do it very efficiently and also in a very quick manner, we rely on computers. I have used here a variety of different machine learning methods to do this. So basically, the idea is to give as an input the metabolomics data set into the machine learning platform in order to, and ask actually the machine learning platform to identify patterns in my metabolomics data and then classify it into healthy and NAFLD. The next slide, please. So here you can see the metabolite classes that have been detected in the different individuals. Their compositions are seen for the healthy and the ones who have the diagnosed NAFLD disease on the left-hand corner. On the right, so when we feed this metabolomics data into the machine learning platform, as you can see in the right, we have different machine learning methods. The pattern recognition from the artificial neural network method performs the best. This is also seen in the confusion plot that when we give the metabolomics data into the artificial neural network platform, it's able to identify patterns from this data, and then it classifies into NAFLD and non-NAFLD with an accuracy of 71.5%. So basically, we are working on improving this accuracy of classification and further validating it on different cohorts so that we could diagnose NAFLD at a very early stage in a non-invasive way. So thank you for your attention.
All right. Thank you, Ambreen. That was wonderful and quick. Bringing in our judges. Over to you, judges, if you have any questions.
Yeah, I have a few questions. What was the source of the data in terms of database and the type of cells and tissues first? And how confident you are of the accuracy when you say, I mean, any method would use some certain positives and certain negative. What was your comparison with?
Okay, to answer your first question, here I had shown you the results of the plasma samples, and these were collected by our consortium partners. And we are also collecting samples from stool, adipose tissue, and so on. So as you can, and to answer your second question, so yeah, you're right that if you go to computers, they are not completely accurate. So that's why we need to test it on different cohorts, and we are doing that right now.
But published literature, any guess, like compared to similar studies, what is the false positives and negatives?
So currently, there are studies which use the same method to actually classify the MRI images. So these are on the rise. And this is a new approach to actually try to classify the metabolomics data, and we are just trying it out. Of course, with an accuracy of 71.2%, we are not going to publish it. We are just trying to increase this accuracy and working on it.
Ambreen, did you say you are an undergrad student?
No, sir, I'm doing my PhD right now.
You do a PhD. Okay.
Yes.
And which year you are?
I'm in my second year now.
Okay. Thank you.
Thank you.
All right, there you have it. Thank you, Ambreen. It was a wonderful presentation.
Thank you very much for the opportunity. Thank you.
Thank you.
How many more presentations do we have? Because the Excel sheet already, this is not present. I just added one more row.
Yeah, this is over now. So all the presentations are over. This was the last. All right. So with this, we come to an end of our poster presentation. And I would like to thank all the judges. It was really wonderful to have you all on board. And yes, it became a little long, more than 3 hours, 15 minutes now. So I would like to thank all our judges who, spent so much time with us, evaluating all of you. And yes, this was the first time that some of you got a chance to come on the other side of our webinars where you are actually presenting and you are being evaluated. It was really insightful and wonderful. So thank you, Dr. Raja, Dr. Satyadas, Dr. Subramanya, Dr. Shashidhar, Dr. Kshitiz, as always, you have been a great support. And Dr. Ramesh, thank you so much for your time today. So people, thank you so much. Audience, I hope you really enjoyed the session today. It was wonderful indeed. I know a little, some of you are disappointed, so don't worry. If you have qualified into the most popular presentation category, we will be contacting you shortly. So we will be announcing the prizes for all the categories, the most popular presentation category, as well as who won today. Okay, so top three, all that will be announced on 28th of September, along with a small experience session by all the poster presenters, and of course our judges will share their experience and how it went all. So with this, we come to an end, and this would not have been possible without the sponsors who have supported us, who have sponsored the prize money, and of course enabled us to do this. I hope you all liked it. Yes. So thank you so much. Now we will be playing a small commercial for our sponsors, C6 Energy and other sponsors. And the judges, I'll meet you at the other end. Thank you.
हमें आत्मनिर्भर बनना होगा। कोरोना के संकट काल में हमने देखा कि बहुत सी चीजों के लिए हम कठिनाइयों में हैं, हमें दुनिया से लाना है, दुनिया दे नहीं पा रही। हमारे देश के नौजवानों ने, हमारे देश के उद्यमियों ने, हमारे देश के उद्योग जगत के लोगों ने बीड़ा उठा लिया। जिस देश में N95 नहीं बनता था, बनने लगे। PPE नहीं बनता था, बनने लग गए। वेंटिलेटर नहीं बनता था, बनने लग गए।
It has been an honor to work with Bharat Electronics and DRDO to deliver 30,000 ventilators to the nation. Scanray is the CV200. Scanray was one of the first companies in the country to manufacture advanced medical equipment and export to about 80 countries. So naturally, the responsibility of delivering ventilators for the nation came on us.
Scanray has been manufacturing ventilators for many years at our Mysore facility. But manufacturing 30,000 ventilators in a short span of 2 months was a great positive challenge. We have replicated our complete manufacturing testing facilities, ramped up the supply chain, streamlined the procurement, and improved the logistics to achieve this.
It was a seamless effort which can take country to the next level of Atmanirbhar Bharat and Make in India, to make India self-sufficient in medtech and enable it to deliver products to the globe.
I really think what you have done should inspire other people in different areas of manufacturing, saying if Scanray can do this, we can also do this.
Scanray is a movement, not just a healthcare technology company.
Humanity now consumes much more than the planet can replenish. In the last 45 years, the demand for Earth's natural resources has doubled. Just feeding the growing population is one of the challenges we will face in the coming decades. The world is experiencing the twilight of oil. Water continues to be the source of wars. The ravages to this planet are seen everywhere. And no, we don't have another planet to go to. Humanity has also showed us time and again that powerful intentions paves way for a new world.